Hello, everyone, and welcome to today's The Scientist Symposium. My name is Nathan Nee, Senior Science Editor for The Scientist, and I will be moderating our discussion. Today, our expert panel will examine immunotherapies and other immune supporting strategies that help prevent, control, and eliminate cancer. Before we begin, I'd like to thank ATCC, Biorad, Canopy Biosciences, Sinobiological, and BPS Bioscience for sponsoring today's webinar. We like our symposia to be interactive. We encourage you to send us your questions or comments at any point, and our panel will address them during the Q&A session that follows our presentations today. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. Our sponsors have provided us with some helpful resources related to today's webinar, and we have posted these in our handout section on the right side of your screen. You can access and download these documents at any time during the webinar. If you have minimized the Q&A panel, simply click on the blue chat button in the bottom right corner to bring it back. This symposium will be archived on the Scientist website, and we will send you the link via email for the on-demand version within a couple of days. Please note that you will not be able to download the presentation slides. With that, let me introduce our first speaker. Joachim Lundeberg has recently focused on spatial transcriptomics, a technique that enables a detailed description of gene expression patterns in tissue sections, and was featured as Method of the Year in 2020 by Nature Methods. Through his work, Lundeberg has demonstrated the development of the technology to facilitate multimodal analysis. He has also, through data-driven analysis of tumors and their microenvironment, provided examples of how spatial analysis can impact cancer research. Let's just make sure everything's up and running from a technical perspective. Dr. Lundeberg, could you please share your screen?
I think I'm sharing the screen already.
And it's not quite popping up on our end yet.
I'll redo it. Sorry about that. So, how does that look like?
I can see it in presenter mode, but not in presentation mode. There we go.
How about that?
Yes.
Okay, sorry about that.
No problem.
Okay.
If you could just hide the bigmarker.com as sharing your screen prompt. Yep, there we go.
Okay, thank you. Thank you for the invitation to talk here today. I'll talk about some of the work that we're doing in the space of spatial transcriptomics to study immune cells and the tumor. So I do have a slight issue that I don't see my...
Yes.
No. Okay, thanks. So just a disclosure, I'm consultant for 10x Genomics. So I thought I'd just give a background to the research we're doing, focusing on spatial biology and how we could address things in space or in tissue, and then see how we could use multimodality to increase the power of such analysis. And then I'll then talk more extensively about how we track tumors and how we study the immune microenvironment in tissues. So we have had a long interest in space or spatial context, and obviously there are enormous, very good tools to study. If you have your favorite gene, you have just design your probe to query where that is based in a tissue using in site hybridization. Or if you have a protein, you could actually start looking for that presence of protein in a tissue. And this type of technology has been extremely valuable in providing the Allen Brain Atlas and the Human Protein Atlas as to describe the spatial context of a gene or a protein. So the issue with this is really the low degree of multiplexing. So that was the rationale for us to try to develop a technology to not study a single gene or a single protein, but to study all expressed genes, the whole transcriptome. And the basic is then to extract the RNA from a tissue section. And by doing that, we realized that we have to get the tools to study all genes being expressed. And we had to develop tools to analyze those. And actually that fed into some computational effort that I will briefly mention in this presentation. And then we apply them in spatial biology. So I will actually make a small tour about this to start and see how we are using these tools to study tumor immune systems. So the work that we started a few years back then resulted in a paper in 2016 by the amazing team of Jonas Versén, Patrick Stoll, Fredrik Samen, and Sanna Vikovic. And this method that we developed at that time was really based on taking any sample, sectioning that, placing a tissue section onto a glass slide, and we stain the tissue, we get the H&E, we get the rich morphology from that, but beneath the tissue section we have a barcoding system, and in each spot we have a poly-A probe or poly-T probe that captures the mRNA through its poly-A tail, but each position on the array also has a spatial barcode. So when we permeabilize the tissue, let the mRNA migrate down to these, we are then generating cDNA. And this cDNA is then put into a sequencing instrument that will tell us which gene we have and which position that has. And since we capture the entire transcriptome through the poly-A tail of the mRNA, we get thousands of genes in each and every spot. And having that resource, we could actually explore the data to see what is gene expression telling us about the tissue context. And we do that on the mRNA, but we also done some work on the DNA, but I will not cover that today. So just to get everyone on the same page, these are then to the left, you see 4 different experiments, and then you have the number of genes that we capture in each and every spot. So these are simply based on the abundance of the RNA present in that spot or beneath that spot. What we generate then is a count matrix where each spot has different expression levels of different genes. And this is very similar to the single cell world. And in that next step, you look for gene expression similarity. So you get your clusters of similarity, and these are then usually colored. But what's different with spatial data as compared to single cell data is that all of these colored spots has a spatial location. So in the end part of the experiment, you bring out the H&E image that you took up front and place the data-driven patterns of gene expression similarity on top of the morphology. And usually the gene expression matches the morphology, but as I will show, gene expression actually provides much higher granularity of the information contained within that tissue section. And as mentioned, Nature Methods said that this type of technology was the method of the year. And on the cover, you actually see one of our experiments with the barcoding glass surface on the bottom. On top of that, you have an H&E stain of small intestine. And on top of that, you have the different colors representing the data-driven gene expression patterns. So the technology that we developed was later then acquired by 10x and are now available as a Visium. And you could start with both fresh frozen FFP samples. So this is a sequencing-based transcriptomic technology. You capture the entire genome, transcriptome, but you should also be aware that there's a lot of methods being developed right now in this space, indicating the large interest in the field of to spatially resolved transcripts. Some of these technologies are sequencing-based, others are based on imaging-based transcriptomics, where you design a probe panel, very much like the in situ hybridization technology, but you have larger sets of panels, hundreds up to 1,000 gene panels. But still, Visium is then the most well-used method right now, because you really don't need any instrumentation to do this more than what you have in an histology lab. So as I mentioned, there's a flood of papers coming out how to look at spatial data, and we have a paper came out a week ago, which we call SEMLA, which is a toolkit for spatially resolved transcriptomics. And this is just capturing different things that you're interested in spatial data. One is being cell type mapping, where you have Visium data and a single cell data, and you could deconvolute which cell types are present in the sample. And I'll give some examples of that as I continue. But you could also look on radial distances. How does gene expression differ at different distances in the neighborhood of a tumor, for instance? And you could look for spatial autocorrelation and several tools for spatial statistics on neighborhoods and similar things. As a very basic example, how we could analyze breast cancer tumors was published some time ago, where we used the pathologies annotation of a breast cancer sample, a HER2 positive sample. And we could then map the different cell types, thanks to that we have single-cell RNA sequencing data. So in this particular data set, we ask where are B cells, and you could get a quantification of that, and then you could ask the question, where are T cells? And then you could combine these in a very simple scoring to score TLS structures, or tertiary lymphoid structures. So that's really an overlap in B and T cells. So this is really handy, especially if you look on what you have on the H&E image. It's really hard to pinpoint, sorry about that. It's really hard, challenging to pinpoint that TLS structure, while on the right side, you actually see how we use this TLS score to search through an entire Visium experiment. Are there any TLSs? So you could actually use this type of informatic approach to look for very subtle morphological structures in the data. So we usually talk about multimodular analysis in general. And what I mean by the conventional one is that you have morphology, which is really rich in information. You could have the single-cell RNA sequencing generated by yourself or in public domain, and then you could use spatial transcriptomics. And this is a study that we did some years ago, but I just want to point to things that actually relate then to the immune response. So this was a collaboration with Paul and Andrew at Stanford, where we had H&E images of squamous cell cancer. And in parallel, Andrew actually generated single-cell RNA sequencing data of these keratinocytes. And he identified what we expected to find, really the cycling cells, the cycling keratinocyte, the basal keratinocyte, and the differentiating keratinocyte. But on top of that, when he took a tumor sample, he also identified a new population, tumor-specific keratinocyte, which we call TSK. So the immediate question is obviously, where are they based? So then you could actually do your spatial experiment. And what we find is that these TSK are enriched in the tumor edge of this. And we just map down those cells. So this TSK then has an EMT program indicating that this is indeed the invasive front of that. But what is clear by this schematic view is also that this is only part of the edge. So we continued the analysis, and we actually sorted out the edge of the tumor. And then we took those and looked for what kind of information they are bearing. So for this patient too, we identified 2 data-driven clusters in the edge, number 5, and when we look at the gene ontology, we actually find that these are immune response genes or involved inflammation, while this tumor edge is actually then more what you expect, a basal tumor phenotype. So what we kind of end up with this analysis is that we have different programs along the edge of the tumor, and one part being the inflammatory niche. And I think this has a bearing in the way that you look on treatment. So in this data set, the single cell data set, we have a variety of cell types, and you could actually then go into the cell types and look for marker genes that you would like to approach by different checkpoint inhibitors. Like up here, it's the PDL1, and you see it's expressed in migrating dendritic cells. And then you have this CD276, which is expressed in this TSK, this new population. So this is really obtained already by single-cell RNA sequencing, but we could actually then identify that these two populations are based at different parts along on the edge. And I think that has some bearing in how you think about the treatment of this. So we have different modes of immune-evasion across the tumor surface. And I'll come back to the tumor age later on in this to just further discuss how that relates to the immune response. So this is really the message of this is that you have to consider the entire tumor, and you have to consider the tumor edge when you start thinking about how to treat those. So we have an ongoing collaboration with Alex Warbrick at Garvan Institute in Sydney. And a few years back, we embarked on the first molecular characterization of human breast cancer. Not the first, but one of the first that combined single cell and spatially resolved atlases of human breast cancer. That was a rather small study, but now we have increased this to more than 300 samples. And this is a Pandora's box of biology, because you have really rich metadata for these 300 samples, like avatars and single cell data and whole genome data. Our contribution to this is really to look into the spatial parts of this. And some of this relates also to genomic variation that I will not cover. So obviously, when we do these kind of large-scale studies, we really would like to combine as many modalities as possible. And if you could do this on the single tissue section, you really are improving the power in the analysis instead of taking adjacent sections, having batch effects and things like that. So these are the molecular barcoding available today, and I'll touch upon two of these modalities. One is the transcriptome, which is really multimodality at the RNA level, and I will then add off with metabolome studies as a segue for next speaker as well, I guess. So on the RNA side, we really would like to understand the cell types in breast cancer, and we really would like to understand antigen recognition or clonotypes. So we do a standard ST protocol. We place our tissues onto these Visium arrays. We do imaging, we collect that morphology, and then we do molecular tricks to get the RNA down and analyzed. But what we end up in the end of one of the days is really a full-length cDNA library. This library is in the standard procedure fragmented, and the purpose of that is to adapt it for Illumina 3' poly-A sequencing. And this is really what I talked about previously, where you just overlay your gene expression patterns over the H&E image, and then you could start looking for your cell types in that data. And short read sequencing is actually what the majority of the cell type annotation databases are based on. So you have a compatibility between the different modalities here. So earlier this year, we showed that we could actually take that full-length library that we generated and do long-read sequencing. And we did this a demonstration using the mouse brain. We could identify then isoforms using Oxford Nanopore, isoforms from the mouse brain. And this was kind of the inspiration for the next project, which is really then to try to pinpoint T and B cell clonality. And what we are interested in here is obviously the variable region binding to the antigen of B and T cells. And this variable region is obviously very variable. And how can we usually analyze that type of data. There's a challenge with this, and this audience are probably well aware of that. You have this VDJ recombination, and then for B cells, you also have somatic hypermutation, and you also have isotype switching. But this is really what you really would like to analyze if you would like to understand the antigen binding. So one could say, just to avoid confusion, that if you have a unique CDR3 sequence, we call them B and T cell clones. So these are then clones that have the same receptor sequence. So, and this is just alluding a little bit to what I talked about previously in the standard protocol of an mRNA. You bind that through the poly-A tail if you're working with fresh frozen samples. You do your cDNA sequencing, and the CDR3 sequence, the VDJ, is pretty far from any of the parts in this protocol. So when you do fragmentation, you actually lose the VDJ region. So only have the constant region of this full-length library. And the constant region is sufficient to say if it's a B and T cell, but we will not tell you the binding properties. So we're missing that in the standard way. So what we developed then was that we generate our full-length library in the tube, and we split that library into two parts. One is the standard spatial gene expression, looking at the 3' end of the mRNA. By that, we could deduce which cell types we have. And on the other side, we are doing a pull down of the B and T cell receptor. And we use array probes that then conserves the CDR3 sequence, but also contains the spatial barcode. And you could read more about this in the bioRxiv submission or preprint. So I mentioned earlier the single tissue section technology, which is what technology? It's the basis is that you just overlap. You have the H&E image, and from the same tissue section, you can get these data-driven patterns. And on top of that, you could identify which clonotypes you have of T cells and B cells. And this work is a big collaboration with a group at Karolinska Institute, headed by Camilla, Jeff, and Kirong, as well as group members from my part. And obviously Jonas Versén, who's the main PI. So developing this technology, we went into some benchmarking first, and one of the early cases, one of the good cases to do benchmark is tonsil tissue. So we did a number of tissue sections. These are fresh frozen samples, and we took these at different layers and used them for different purposes. So we did bulk SmartSeq VDJ sequencing, and then we did spatial gene expression and our spatial VDJ on some sections, and we compared that with other validation like RNA-scope and things like that. So when we look at our data, we get the big families of B cells and T cells families, and in total we identify over 60,000 unique CDR3 clones. And when we do the 3' sequencing, we have identified some 14,000 genes. So the pull down is really efficient for this analysis. So then we could actually then compare that with our bulk data, and we have an overlap between 30 and 50% of clonal sequences. And that's a pretty good number, as you have to remember that these you have a lot of single CDR sequences that only occur once, and the probability that you find that in bulk and in space is in spatial data is pretty small. So we were super excited by this data. So then we could actually then start looking into the spatial dimension of these clones. So what you see on the top left is just an overview. Where do we find these clones over the tonsil tissue space? And you could see some patterns, and I'll come back to that. And then you also have this T cell clone distribution, and you see them also all over the tissue. But there's a lot more information about this, and I'll show you shortly. So we could then dig in and study specific clones. So this is a B cell clone, and you could actually see that all the counts we get for this unique sequence, which has this amino acid, is spaced very close together. While on the other hand, when we look at T cells clones, they are more spread out over this and actually outside this structure, which is the germinal center of tonsil. So this is just 2 clones and their distribution over space. So now we could start using all these different modalities. So what you see on the left here is the data-driven patterns of gene expression. And you could actually then see different regions here. You could see where you have B cells, and they actually then overlap with the morphology of the germinal centers, the follicles. So that's where you find the B cells. You have a T cell stone, which is outside those B cell follicles. And then you have some endothelium, and you have some squamous epithelium up here. So we could then dig in, because we have the data, we could identify which cell types. So these are 3 panels looking at B cells, plasma cells, and T cells, and where they are based. And here you also see some marker genes for those different cell types. So we could actually clearly see that the B cells are really based in the germinal, in the follicles, in the germinal centers. The plasma cells are on the outer surface here of the tonsil, and T cells are in between these germinal centers. And this you could also validate for some of these marker genes for these 3 cell types. And then we validate that with RNA-scope, and that makes a very coherent view of that we have some good data. We have an interest in general, not only to make a 2-dimensional snapshot of gene expression over a tissue surface, we have a deep interest in the 3D perspective of these samples. So this is just exemplifying that, what you could achieve, and just look for one single clone. This is clone 30, which has this sequence, amino acid sequence. And here you see section 12, sections 16, 17, and so forth, and the distance between those sections. And now we're looking at one single clone through those different tissue sections. And you could clearly see that they are all in the same germinal center throughout these distance in 3D. And I think this give us a flavor of the size of the germinal center, but also the localization of a single clone. So having these sections, we could then start annotating in a very trivial way numbers 1, 2, 20, 36, 37. And we could then follow these germinal centers through this dilution series. And then we can start looking at how many clonal family counts we have. And we could simply identify that the largest area here has the highest count, which is not that surprising. But you could actually see there's a variation between these germinal centers. So the question then is one IgG clone restricted to one follicle? Well, I showed you these consecutive sections, and it was really one clone in one follicle. And that's true for most of the clones. Most of the clones are restricted to one follicle. And this is exemplified here. You could actually see these are 3 different clones, and they're all based in one single follicle in a very dominant way. So these we called restricted. And you could actually see here, this is another germinal center, and this clone is based here, which is one area, which is very consistent. But some clones actually have more than one dominant follicle, and this is just showing that these are the different annotated follicles and the number of non-restricted patterns, how many, where the unique clones are distributed. And you could actually see it over here that these are then localized in multiple follicles. So what's so exciting now, I think it's exciting, is that you could actually use the somatic hypermutation to mark also some clonal perspective of evolution of these. So we could actually, using that information, we could start looking at phylogenetic relationships. So what you see here is I have two screens, so I'm sorry about the confusion of my pointer. So this is a single clone family, and this is the unmutated ancestor that we don't see, and then you could see all the clones that we have see the relationship using this somatic hypermutation. And I would like to stress, if you look on number 22, it really is the starting point to a completely new diversification or evolution of new clones. So we could take this tree and go into space or spatial and see how that looks like. So this is a simplification of that lineage tree. You see the green here, and how that, this is actually then the 22 that gives this new lineage. And when we look into space, this is the green clones here, and that part, and you could actually see the structure of the germinal center here. And here you see number 22 that I just mentioned, which is coming just outside the germinal center. And you could also imagine how that then seeds into another and expands. And I think this is a beautiful view where you combine evolution with clonal types. I have then a particular interest of marrying this kind of analysis with the breast cancer project that we are ongoing. And this is just to exemplify the power of this. So this is a breast tumor again. We have two patients. We're looking at 4 different regions here. This is just one region, region A from patient 2. And this is what the pathologist is indicating. This is where you have invasive cancer, and this is where you have stroma. So now we have a fantastic tool set then, because we could use all of the tools that I just talked about. The first part is to study the tumor. And we could identify 3 different gene expression programs, and we call them tumor clone 3, 5, and 8, and they are distinct in gene expression, as shown on the left side. Sorry about that again. So what you see here is that tumor clone 3 does not overlap with tumor clone 5, and tumor clone 8 is just on the side. So we actually have 3 different activities of the tumor, and then the obvious question: how does the B cell repertoire look like? So you could actually see here, this is clone 0 and this is clone 39, and you could actually see that they are distinct. So this is a unique binding sequence, and they localize at different parts. Then we bring in our information about the tumor clones. We could mark out the border or the tumor edge that I talked about earlier, and then we could marry these two into analysis. And I think this is so cool, because what you see here is the clone, the different unique sequences, and they're all placed at the border for the tumor clone 3, and this is also true for tumor clone 5. The clones that we identify having a unique CDR sequence is on the border. So I think that is very empowering to study the tumor, but also getting a feeling how the immune cell repertoire evolves with that. And I think we are also having similar data on how the tumor genome evolves, but I haven't the time to look at that. So combined, we could do quite fascinating studies of the pseudotemporal ecosystem of tumors. But I just would like to add that what we could do today is also to study the metabolome at the same site. This example was just published a few weeks ago. It's not breast cancer, but it kind of proves the point that you could actually think about this. So using mass spectrometry imaging, and this is what you see up to the right, that you let the laser go through the tissue, and in each spot you collect a mass spectra. And then you compare these peaks you get from this analysis to different metabolites, lipids, glycans, or neurotransmitters. And you could imagine this laser would destroy the RNA in that tissue section that it just passed. But we challenged that, and we took a brain sample, we put that onto these arrays, we cover that with different matrices in order to be able to look for different components of the metabolites. And this is looking at the neurotransmitters and some lipids. Then we are taking that single tissue section and we do multi, as displays up to the right. We wash away the matrix, we get the histology, and after that, we continue with the spatial transcriptomics. So we actually get this kind of layered information from the same tissue section. And the power of this is multimodality by comparing the correlation of different measurements. And this is my favorite image of that paper, which is supplementary 54 or something like that. And what you see here is a small cluster of mass spectra that correlates with the number of genes. This allows us then to look at very subtle changes, and this is from a Parkinson model with very small changes, but it picks out new metabolites and new genes in the context of Parkinson. So this is really powerful. Unfortunately, I missed checking my time. So if Nat could just indicate how much, do I have anything left?
I think another 5 minutes is fine.
Okay, very good. So, so anyone that is doing Visium knows it's kind of expensive to run these experiments. So I'm a strong believer of deep learning. I have this amazing student, Ludwig Beinstråle, that last year published a paper that actually marries deep learning and spatial transcriptomics data. And what you really can now foresee is that if you have rich morphology, which is a prerequisite for deep learning, is that you could think of a stack of tissue sections of a tissue that you would like to interrogate. And then you do your spatial analysis, you collect your spatial data, your morphology data, your gene expression data for 2 tissue sections on this stack. And you put that into a deep learning model. But if you then also collect the H&E image, it is possible then to impute the gene expression solely based on the H&E image. So in a way, you can, by 2 experiments, you gain 4 for free, more or less. And this was then shown in this paper. And I think this is just an appetizer of what is to come, and kind of proves that we could actually use deep learning in a very cost-effective way. So with that, I'm closing in. I think I'd try to show that we could actually link tumor cell clones with specific B cell and T cell clones. I think this is very interesting. We could use data-driven approaches to annotate tissue to guide us to do the right choices and not only depend on histology. And we could generate single-cell resolution by this deep learning that I talked about, or expansion microscopy. And the tools that are out there are expanding very, very rapidly, and it's hard to choose really. But I think this will calm down eventually. But I think my main message is the power of multimodality. If you have spatial transcriptomics data, you could infer cell types in your data. You could actually infer genomic variations looking at clones. You could look for small molecules by mass spec, and you could actually get these immune clonotypes. And if you combine that with morphology, you really get a good chance to resolve the cancer ecosystem in a very powerful way, I think. With that, I would like to thank the funders and also my collaborators. As I'm a technologist by training, I'm very much dependent on people that I collaborate with. And in this particular case, I would like to highlight Alex for breast cancer, Jonas and Camilla for this clonal evolution of clonotypes, Pandrian for the SMI, and I mentioned also Paul for the squamous cell carcinoma. With that, yes, I need to also mention that Reza, Alma, Kim, Ludwig, Saneta, Ludwig, Michel, and Marco all contribute to the work that I talked about today. With that, thank you.
Thank you very much for that presentation, Dr. Lundeberg. At this point, let me introduce our next speaker. Arun Vita is a physician scientist and associate professor in the Department of Laboratory Medicine and the Department of Bioengineering and Therapeutic Sciences at the University of California, San Francisco. He completed his residency in clinical pathology at UCSF and his MD and PhD at Columbia. Vita researches mass spectrometry-based proteomics, target discovery, protein engineering, and cellular engineering towards the development of novel immunotherapies for hematologic malignancies, while his clinical duties center on molecular genomic pathology. Vita also directs the UCSF Stephen and Nancy Grand Multiple Myeloma Translational Initiative Laboratory, and is a prior recipient of the NIH Director's New Innovator Award, a Clinical Scientist Development Award from the Doris Duke Charitable Foundation, and a Chan Zuckerberg BioHub Investigator Award, among others. So let's just make sure everything's up and running from a technological perspective.
All right, so go ahead and share. Let's see. Hopefully that's showing. Let me start.
Yep.
Oh, sorry, Rose, I'm not on the title slide though. My fault. Okay, is that the correct view?
I'm getting the presenter view, not the
Okay, let me
presentation
try and
view.
let me swap it.
There we go. Perfect.
Hi, see it now? Okay,
Yeah.
great. And let me pull up my pointer if it wants to show up. All right, great. Thanks everyone for attending today, and thanks so much to the scientists for the invitation to discuss our research here at the University of California, San Francisco. That was a beautiful talk from Dr. Lundeberg, and very difficult to follow up. Amazing technology that he discussed in terms of being able to characterize the immune microenvironment in cancer. What I'm going to be talking about today is a different theme within immunotherapy, really getting much more toward therapeutics and discovery of novel targets and development of new therapeutics. For me as a physician scientist, this is definitely something that's near and dear to my heart, is actually trying to make new drugs to directly benefit patients. So I'll briefly list my disclosures here. This is going to advance. Let's see, try down here. Okay, so I'll list my disclosures. So what I'm going to be talking about today, in terms of cancer immunotherapy, there's a few different modalities that you may hear about that are out there. You may be familiar with cancer vaccines or checkpoint inhibitors. What I'm going to be talking about today is other classes of immunotherapies that are really quite broad, that focus on biological molecules that selectively recognize and target antigens on the surface of tumor cells. And so what do we mean by this? Well, there's many molecules that you may have heard of, say rituximab, for example, that's been around for over 20 years. These are so-called naked monoclonal antibodies that can target, in this case, CD20 on the surface of B-cell cancers. You have newer molecules that have been FDA approved more recently, bispecific T-cell engagers, where one arm of the antibody targets an antigen on the surface of a tumor cell, say CD19 and other B-cell cancers. The other arm targets CD3 on the surface of a T-cell. This essentially creates an artificial synapse so the T cell can kill the tumor cell. And then others such as antibody drug conjugates. We have these approved for several targets, say in hematologic malignancies, for CD33 in acute myeloid leukemia, where you have an antibody that recognizes an antigen on the surface of tumor cells that's conjugated to a toxic warhead that then internalized and can kill the tumor. What my group mostly focuses on are engineered cellular therapies. So sorry, I'm trying to get back here. So within engineered cellular therapies, these are an approach where essentially you can take a patient's own immune cells or potentially a donor's immune cells and genetically engineer them to express an antibody fragment on the surface, whereas internally they have some part of the native activation machinery for that immune cell. You again create an artificial synapse, the tumor cell in which case here now the antibody determines the specificity. You're no longer dependent on, say, the TCR, for example. This antibody fragment binds to this tumor cell surface, this tumor surface antigen, and then leads to the killing of the tumor by the T cell. Most advanced in this space are CAR T cells, or chimeric antigen receptor T cells. I'm going to be telling you more about that today. The CAR NK cells and even CAR macrophages are in development now. And the one thing all these therapies have in common, I mentioned, they're all essentially based on antibodies that recognize a tumor surface antigen. And when it comes down to this, all of these therapies, to be useful, really need a good surface antigen. And so what do we mean by that? I'm going to get to that in just a slide. Before that, though, I am going to tell you why we focus on cellular therapies in particular. And that's mostly what we're going to be talking about in the talk today. So this data started coming out several years ago using so-called these chimeric antigen receptors, or CAR T cells. This is an example in B-cell lymphoma patients that were extremely refractory to all of the therapies, previously with less than 10% expected survival. CAR T cells targeting the cell surface antigen CD19 on these tumors could lead to cures, and these have proven to be durable, even a longer follow-up, in approximately 50% of these patients. And so this is really remarkable, really showing that with a single dose of this therapeutic, you could get cures. This really has not been demonstrated for any other therapeutic modality. It really got my lab to move into this space, and something that's quite exciting. And we also have very strong patient advocates, such as Emily Whitehead, who was treated by Carl June and others at the University of Pennsylvania now, I think over 12 years ago. Single dose, her pediatric B-cell leukemia was cured. So really something that makes this modality particularly interesting and exciting. And so we're thinking about CAR T cells in particular. These are a very potent therapeutic. And so in coming back to this question of what makes a good surface antigen, essentially, ideally, what you would find is on these cancer cells, an antigen that's expressed at high density, uniformly, essentially having this high amount of target on the cells, much higher levels than you find on any normal cells, where normal cells, either these antigen is either totally absent or expressed at very low levels. And then also optimally on the tumor cells, it's expressed quite homogeneously. You don't have inter-tumor heterogeneity, where essentially maybe some tumor cells are negative for the antigen and won't be eliminated by any therapeutic targeting. This turns out to be a really big challenge for many types of cancers. I'll talk more about some of the pitfalls we've seen with solid tumors on the next slide. In blood cancers as well, we've had the most success with targets called CD19 and BCMA. Both of these are on B-cell derived tumors, either lymphomas, leukemias, or multiple myeloma. And essentially, in those cases, we're kind of lucky in that those antigens are expressed on the normal cell counterpart, but patients can actually live and tolerate a lack of normal B cells, lack of normal plasma cells, if we supplement them with exogenous immunoglobulins therapeutically. And so, but for other tissues, that's not the case. For the example from Dr. Lundberg's talk, for breast cancer, for lung cancer, for colorectal cancer, those normal epithelial tissues, for the most part, are considered essential. They would lead to severe toxicities if we also target them, even with a tumor upregulated antigen. And so this is really one of the big challenges in terms of target discovery is, can we find these good or optimal targets? And what are the strategies we can use to do that? One of the main reasons we think about this quite a bit in my lab, and definitely regulatory agencies such as the Food and Drug Administration in the US think about this very heavily, is this idea that particularly CAR T cells, since they are so potent, they have the ability to eliminate cells that express several dozen or hundreds of copies of this antigen on the surface per cell. So even if you have a tumor cell that expresses several thousand copies of antigen, this is great if your CAR T cell is eliminating these tumor cells, but this can lead to these severe toxicities. And I think one of the most important examples is a case report now from several years ago from the group at the NIH. What they found was that they made initial CAR T cells targeting HER2, the gene name is ERBB2. They make CAR T cells targeting HER2 for patients with, in this case, for patients with colorectal cancer, I believe, in this specific report. And what they found was that patients actually died due to toxicity, due to low levels of HER2 on lung epithelium. And so this is really something we know can lead to severe toxicities. This has been seen in other contexts too, of the so-called on-target, off-tumor tox. Is something that we really think about. And so this has also been seen for other solid tumors, for issues with EGFR, GD2, other targets seen in other solid tumors. So the question is, how can we get around and address these issues? So in terms of new target discovery, The strategy that's really used the most in the literature in the most recent years is thinking about transcriptional analysis. And this is an example from a colleague of ours, Eric Smith at Dana-Farber Cancer Institute. He had this really nice paper which he found using transcriptional analysis, examining the blood cancer multiple myeloma. They looked at expression of all genes across the transcriptome and found that specifically this transcript GPRC5D, which encodes an orphan 7-pass transmembrane receptor, no known function, still don't really know what the function is, but appeared to be very highly expressed in myeloma tumor cells compared to all other cancer cell types in the Cancer Cell Line Encyclopedia. They went on to compare this to normal transcriptome databases in the Genome Tissue Expression Database, or GTEx, finding that the myeloma cells had much higher transcript expression than all these normal tissues, except for some expression within the skin. But this was deemed to be an acceptable potential on-target, off-tumor tox profile. I'll say that these therapies targeting this antigen have now moved into clinical trials and been very successful. A bispecific therapeutic is now FDA approved in multiple myeloma. But interestingly, this was predictive. The main off-target tox is actually in the skin and nails. And so, so this approach to looking at the on-target off-tumor tox did actually show what ended up happening in patients. And so this transcriptional analysis is powerful. I think GPRC5D is one of the best examples of what's come out of those approaches, because transcriptional databases are much more accessible. We have these large-scale tumor-focused or normal tissue-focused transcriptional data sets. And so many folks have looked at this. The issue is now at this point, kind of progress in this area has been relatively stalled. Kind of what's out there to find, the databases have been mined, they've been a bit exhausted. And so the question is, can we go beyond this? And so what we try to work on in my lab is strategies. I lost. Okay, there we go. Is strategies to think about beyond the transcriptome. One of the main issues is that regulation of proteins at the tumor cell surface, which is what we're really interested in this context, can be quite complex. And so whether going from in the nucleus, you're making the transcripts, these can be distributed in other parts of the cell, but thinking about going from a transcript to a surface protein, you have translational regulation within the ER, folding dynamics, PTMs that could be put on them, trafficking through the Golgi. Once you're at the cell surface, you can have complex series of endocytosis or antigen shedding. So can we focus on these proteins at the cell surface, especially knowing that mRNA abundance, many studies have shown this, and data from our lab and many others, is not necessarily predictive of surface proteins. And so can we focus on this protein at the cell surface? So the technique that we use to do this is mass spectrometry-based proteomics. Many of you may be familiar with this. Essentially, this is a strategy by which in biological systems, and it has primarily been used for biological discovery work much more than anything else, but by taking complex mass spectra of a sample, looking at thousands and thousands of spectra, we can identify now routinely, say in a cell line culture, other aspects, we can routinely get to depths of 10,000 or so quantified proteins. We can look for post-translational modifications with different routines. We do a lot of this in my lab. This is just one example from a paper where we're looking specifically at extracellular vesicle shedding and their contents. Mass spectrometry is incredibly powerful in this context for biological discovery. And I think what I want to point out in particular is in an unbiased fashion, because now there are proteomics technologies out there that can get to relatively high depth for surface proteins, say with DNA barcoded antibodies and approaches like CytSeq or like cytometry by time of flight or CyTOF. Those approaches can get to several dozen, maybe even several hundred markers. But the issue is you have to know a priori what are the targets you're looking at. Those are all antibody-based. Mass spectrometry still can, and you need to have high-quality antibodies. Mass spectrometry can find targets that were unexpected. And so we think that's really the power here. The approach we use to do this is so-called cell surface capture. We didn't develop this ourselves. This was initially out of Rudy Aebersold's lab in Switzerland in 2009. We've adapted a bit in the lab, but we try and apply it to specific clinical questions that we're interested in. And so in this approach, essentially, it really relies on the covalent biotinylation of cell surface glycoproteins. About 85% of cell surface proteins are thought to be have some form of N-glycosylation. We're able to essentially through an oxidation and then covalent reaction approach, be able to install a biotin on the cell surface proteins. This we can use as a chemical handle in the context of we can then lyse the cells. Ideally, the intracellular proteins do not have this biotin handle on them, whereas our extracellular cell surface proteins do have these biotinylation sites. We can use streptavidin beads to pull down the biotinylated proteins. And then by doing this, we can then take this onto the mass spectrometer, get our tens and thousands of mass spectra, apply various bioinformatic processing to try and see, hey, what is upregulated in our context of interest, what's changed, what's different, what's most abundant. And so this is really, we think, again, a powerful discovery tool to really focus on the tumor cell surface. And so the first part of the data I'll get into, I just want to talk about, is this so-called surfaceomics approach actually useful? And so I'll give an example that's led by a study we published a couple years ago now, but building on work that we're still actively engaged in. Matt Nix was a postdoc in the lab. And what he wanted to do was specifically try to look for novel targets in this poorest prognosis subtype of B-cell acute lymphoblastic leukemia. And for this talk, I'm not going to get super into the background of the different cancers I'm going to talk about. Just to say, my group focuses on blood cancers. I'm going to be telling you about a few different blood cancers. But the idea here was these were patients who are not well served by current therapies, even for CD19 targeting CAR T cells, which can be effective with these patients. They do very frequently relapse. The question is, could we find additional antigens that were upregulated on these tumors? Because we do know that from other data, CAR T efficacy, like most of the immunotherapy modalities I mentioned in the introduction, the efficacy does scale with the density of the target on the cell surface. So finding something upregulated, we thought could have clinical utility. What we did in this context is we performed cell surface proteomics across several different models of B-cell cancers, and then focused on those that were upregulated in this MLL rearrange, also known as KMT2A rearrange is the newer gene name. We found about 60 proteins out of the 800 that we isolated and quantified. We went through several different rounds of trying to filter for that on-target off-tumor tox. Most specifically, I'll come down here just to mention, we focused on genes that had very low expression in normal tissues per that GTEx transcriptomic database. Again, here we did have to turn to the transcriptome because we don't have broad-scale surface proteomic databases across many normal tissues. So looking at transcriptional data is the best we could do in this context. And then we could also go to transcriptional databases for normal hematopoietic cells. In this case, we want to avoid expression of hematopoietic stem and progenitor cells, as well as T cells, because that can complicate our manufacturing and potentially lead to toxicities as well when treating with CAR T cells. And so we ended up discovering this antigen CD72. It had never been described before as a potential therapeutic target in B-cell malignancies, even though in the murine literature, it was quite known to be a B-cell marker that had been used in autoimmunity studies, for example. We went back and just showing some of the data from the human blood atlas, which is led by Dr. Lindberg's colleagues in Sweden. What they found in that database, they included many different data sets within this is part of the human protein atlas. But what we found is that, again, a very high expression in B cells of CD72, but minimal in other hematopoietic tissues, and really no expression in any non-hematopoietic tissues. I'm not showing you here. So it's all very encouraging for on-target off-tumor tox. What's our next steps? Well, now we did this on cell line models due to input requirements for this technology, which I'll tell you about we're working on overcoming. But we have to go to primary sample validation. So we work with our colleagues, Elliot Stieglitz and others at UCSF, to obtain primary B-cell acute lymphoblastic leukemia samples. We could perform flow cytometry for this antigen. We could find that indeed, again, just kind of the cusp of significance here, within our specific genotype of interest, we did find CD72 upregulation compared to some other samples. We also showed this in other contexts. I'm not showing you all the data. And what was also important, what we showed in the paper, is that this target was also present at all other tested primary B-cell acute lymphoblastic leukemia and B-cell non-Hodgkin's lymphoma samples that we looked at. So while it's specifically upregulated in this one poor prognosis genotype, it could have advantages across B-cell malignancies, which we thought was quite promising. So then I did mention to you briefly in terms of we're very interested in these cellular therapies in terms of getting to a real therapeutic modality. So just to get a little bit deeper into how these are designed, essentially the reason these are called chimeric antigen receptors is because we have parts of the native T cell activation machinery, the CD3 zeta domain that's kind of attached as part of the TCR complex, co-stimulatory domains that come from other important proteins within the T cell membrane that lead to additional signaling to activate T cells. And then we have both hinge and transcription regions, membrane regions that link to this antibody fragment on the extracellular surface. And the vast majority of CAR Ts, including 5 of the 6 that are FDA approved, use a so-called single chain variable fragment. Essentially, this is that hypermutated part of the immunoglobulin that actually leads to specificity and recognition of an antigen. In this context, though, we wanted to try and see if we could take a different approach going beyond scFvs for antigen recognition, because these actually do still have issues where you can get CAR-CAR dimerization. These are heterodimers. They can lead to unwanted signaling at the T cell surface. They can lead the T cells to become exhausted, less therapeutically effective. There's other evidence, and the most recent CAR T cell approved in multiple myeloma actually uses this different technology that we thought was quite promising called nanobodies, which I'll tell you about on the next slide. Essentially, nanobodies, they're very small protein fragments. They're essentially half the size of one of those scFvs. They're derived from heavy chain only antibodies that you find in camelids and other evolutionary ancient organisms like sharks and lampreys. But essentially what these nanobodies can do is they can get the same affinity, same target specificity, but they're much more modular. They're obligate monomers. They don't lead to these unwanted heterodimers. They can have very favorable properties in CAR Ts. And in multiple myeloma, the most recent CAR T that was FDA approved, much more efficacious than all those previously investigated, is based on nanobodies. And so we think that they have a lot of promise. At this time, when we were first working on this project, our colleague Ashish Manglik at UCSF, he had recently described this new fully synthetic, computationally designed nanobody library displayed on yeast. All prior nanobody-based CAR Ts had all been performed and optimized through llama immunization, but we wanted to see, could we take the opportunity to do something fully in vitro and demonstrate that in vitro synthetic libraries could generate potent CAR T cells, which hadn't been shown before. And so in this context, we worked with Dr. Manglik's lab. We were able to perform screening of recombinant CD72 by panning them over these complex libraries of nanobodies displayed on yeast. And basically, after several rounds of enrichment, find nanobodies that were very selective for CD72. And I'm essentially going to skip over all this data because, again, this paper was from a couple years ago. I just wanted to introduce the concepts. But basically, what we found was that we could generate CAR Ts. Here showing you a murine model where the brighter glow is more tumor inside the mice. This is using a patient-derived xenograft model, which is considered the gold standard in these preclinical studies. And what we found is that our CD72 nanobody-based CAR was performing quite equally to a gold standard FDA-approved equivalent CD19-based CAR T for these tumors. And we saw a nice survival in these murine studies. And so this was very encouraging for us. What we've been doing the past couple years since this paper came out is really thinking about how do we turn this into something that we can really put into a human in the context of a clinical trial. And so Will Temple is a pediatric hematology oncology fellow working in my lab. One of the strategies that he took along with Matt was saying, well, we know that these are still, even those were computationally derived, they weren't from llama immunization. The backbone of that nanobody was still based on a llama immunoglobulin framework. And so one of the questions was, could we essentially, there we go. Could we essentially change some of those residues in the backbone of the nanobody to human, potentially to lead to something that's less immunogenic once we turn this into a therapy? Because this has been known in the context of other CAR T cells that use murine scFv fragments. Some patients become resistant because they actually develop reactivity and immunogenicity against those murine sequences in the backbone of the antibody fragment. So what Matt and Will did, they generated a series of about 30 different combinations of these amino acids in magenta, where essentially these ones we knew we could change from llama to human and potentially still have proper folding of the nanobody. There are a few that we cannot change. These are related to the obligate monomer properties of the nanobody. So these ones would, if we change these to human, they would dimerize, which is what we wanted to avoid. But so we could do this protein engineering. And what we were surprised to find was that within this humanization approach, we just thought essentially they would all have similar activity, but just be more human. But we actually found a very wide range of activities, showing you in vitro cytotoxicity data, where essentially we could treat these tumor cells in vitro with CAR T cells. Here's the lysis of the tumor cells on the y-axis. This is kind of a reverse dose response, where this is the highest dose of CAR T cells. This is the lowest dose here with none. And what we found was that our parental clone we described in the prior paper, MBD4, we found some humanization variants that had similar activity, but many others really depleted the activity, and some actually were very similar to our negative control of an empty CAR that had no antibody binder at all. So we're really surprised by this phenotype. And this is something that's very much of interest to my lab, is thinking about the biophysics of why this happens, why you get such different phenotypes in these contexts of these very subtle protein changes. But I'm not going to get into that today. What I am going to tell you about is that the one therapeutic candidate that we homed in on is this one called H24, which is a variant that had actually all of these amino acids mutated to human that we have in magenta here. Again, showing this in vitro lysis assay against this mantle cell lymphoma cell line. This H24 had improved lysis at all tested doses. And then here he's showing you an in vivo study, which is a low dose, we call a stress test in vivo of these studies with this very aggressive B-cell leukemia cell line. And what we found was that the H24, again, outperformed other clones that we had been evaluating, including that parental cell line. So we thought that this was quite interesting in terms of finding a variant that, based on humanization, hopefully will have both less immunogenicity but also more potency than our originally described nanobody. And so again, skipping over a lot of the data here, but really just showing you where we want to go with this. Essentially, one thing I have to warn you is that if you get into the CAR T space, you have to tolerate a lot of automotive puns. And so I'm going to subject you to some of that here, in the sense that here we're calling these nanocars, is named after those who may be familiar. Tata in India came out with this nano several years ago, so it's tiny car. So our CD72 nanocars, again, we're trying to be on the road to the clinic at UCSF. So what's the process here? So this is one of the reasons I think getting into the cellular therapy space is very exciting, because you actually have the opportunity to really develop a drug academically and bring it to clinical trials. Our center is not the only one in the US that has the capability to do this. And so what we've done so far is we've optimized this therapeutic. Work I'm not showing you, but this binder and the backbone. Right now, we're doing all the clinical grade manufacturing that we need, in vivo, in vitro cytotox, or toxicity and efficacy studies, pharmacokinetics, biodistribution, writing our clinical trial documents, and plan to submit investigational new drug application to the FDA, hopefully by the second half of next year. Once that gets approved, start a phase 1 trial here at UCSF. And so I've been very fortunate to get support from several different agencies to do this. It's not cheap, I'll tell you that, but it is possible. And so that's part of the message I want to get across, is that if you get into this space, there really is that potential to develop new therapies. For me, myself, I'm learning a lot about drug development as someone who hasn't been in this area before. And it's very exciting to potentially be able to bring something to patients. And within that, I think that's what I want to talk about is, in terms of it, there's really illustrating the goal in the lab is really kind of go the distance. And I think for those of you who watch baseball, my beloved Philadelphia Phillies hopefully will be punching their ticket to the World Series in a few hours. This is their ace, Zach Wheeler. So if Zach Wheeler goes the distance, that means pitching all 9 innings and winning the game. That's a complete game. And so I like to think about this in our context. Can we do this in terms of using proteomics-based approaches to discover new targets, do the appropriate protein engineering that we need to find these antibody fragments, do the cell engineering we need to develop a safe and efficacious therapy, and then move this to clinical translation. And so I like to think about this being essentially the academic physician scientist complete game, which I thought about yesterday as I was making this talk. So I'm going to trademark that right here in front of you all. So just to give another example too, now switching gears about what we can do with cell surface proteomics. This is a paper we published last year where we were looking at cell surface proteomics to try and find new targets in multiple myeloma, that other blood cancer that I mentioned to you. There's still need for new targets. The current therapies are quite effective, but essentially all patients still relapse even if they get progression-free survival of 2-3 years. And so the question is, can we find new targets? We perform cell surface proteomics led by Ian Ferguson and Bonel Patino in my lab. And what they found was essentially by doing cell surface proteomics, integrating with several other bioinformatic databases, looking at proteins that were predicted to be at the cell surface based on gene expression and protein homology, specificity for malignant plasma cells where myeloma is derived in the Cancer Cell Line Encyclopedia, Human Blood Atlas, and then again, going back to GTEx for this human tissue specificity, we ended up with a scoring system. What we found was quantifying over 1,000 surface proteins in this context in our myeloma cell line models, we found the top scoring was BCMA and its closely related homologue TASI, which this is the FDA-approved most common target in multiple myeloma, so it's useful as a positive control. But we're very interested to find several others that kind of seem quite promising in terms of on-target, off-tumor tox profile, potential for efficacy. We specifically worked up this one chemokine receptor called CCR10. What we developed was proof of principle CAR T cells. So Benel and Corinne Kassep in the lab, working with Justin Eckham's lab at UCSF, we developed CAR T cells that were proof of principle, where essentially CCR10 is actually interesting. Instead of needing to generate a new antibody fragment, there's another strategy you can use for certain targets, certain classes actually have a natural ligand, which you can use to engage that receptor if there's a real one-to-one specificity. So we actually made proof of principle CAR T cells using CCL27 that we could target against myeloma cells and kill them in vitro. We could actually also look at, from the proteomic data, one thing you can get is how abundant are these antigens. And so we could actually look at what was the average intensity of different antigens in terms of how frequent they are found in the proteomic data set. Basically, we think this is useful for designing new strategies of creating very strong synapses between CAR T cells, potentially pointing to new therapeutic vulnerabilities, maybe not for immunotherapies, but even for small molecules, by looking at what are the most abundant antigens on the tumor cell surface, which you really couldn't get from any other technology. And then also we can look at, in the context of what about drug treatment? In this context, we could take tumor cells, treat them with a small molecule, and see what changed in the surfaceome. Could we think about new combination therapies for cancer? So in this context, when we treated with a first-line small molecule called lenalidomide, we found that several antigens were strongly upregulated, particularly this one called MUC1, which is a heavily glycosylated surface protein that's been studied in other anti-cancer contexts. We're fortunate to collaborate with Avery Posey at UPenn, who had developed anti-MUC1 CAR Ts. And we could see in this context that lenalidomide did make MUC1 go up even on primary plasma cells. So that was validating the cell line model. And we could co-treat with lenalidomide and these MUC1 CAR T cells and see increased lysis in this context. And so then again, this kind of illustrates this concept of, can you come up with these dual targeting therapies by using the cell surface proteomics to guide you and what targets you want to combine with small molecules. And then finally, we are making progress in applying these approaches to primary tumor samples as well. That's something that we're in very active area investigation in the lab. This is just some additional initial data from that study. So in the last few minutes, what I want to tell you about is kind of, can we go beyond surface protein abundance to find new targets for cancer? And so in this context, we were really inspired by this paper that came out several years ago that suggested from a hybridoma screen, again, this is a totally unexpected finding, basically trying to find antibodies that bound to tumor cells, not to normal cells, using mouse immunization approaches. This group in Japan, what they found was that they developed an antibody that bound specifically to myeloma cells. They didn't know what the target was. They did target deconvolution and found that it was an antibody binding to this open and active conformation of the cell surface protein called integrin beta-7. If you just looked at a transcriptome expression data set, for example, integrin beta-7 doesn't look like a great target because it's expressed on many other hematopoietic cells. You think it's going to have too much of that on-target, off-tumor tox I talked to you about. But what's interesting in this context is the vast majority of normal peripheral blood mononuclear cells have integrin beta-7 in this closed, inactive conformation. Only when T cells get activated, or what they found was myeloma cells constitutively have this active conformation, you can actually have an antibody that binds specifically to this active epitope. And then what they demonstrated is they can make quite potent CAR Ts out of these, and these are moving into phase 1 trials in Japan. And so we're really inspired by this, but the question was, this was a totally random finding. They didn't mean to find a conformation-specific antigen, but it led to this hypothesis that these cancer-specific surface protein conformations may actually be common, again, with this idea that maybe between normal cells and cancer cells, whether due to aberrant tumor metabolism, signaling, other features, maybe the cancer cells are driving these proteins into different conformations. And even by doing abundance proteomics, as I talked to you about before, we may not be able to identify these targets. So the question is, these may be there, the hypothesis is raised, but we need to develop the technology to find them. And so in this context, we did, I'll say we applied this to another blood cancer called acute myeloid leukemia, really dismal prognosis, less than 30% 5-year survival, second most common blood cancer in the United States, so really high unmet medical need. And really importantly, there's currently no approved CAR T cells, really no optimal immunotherapy target known for this disease. So really important opportunity to try and find new useful targets. And so And this work led by Kamal Mundal in my who was in my lab, who's now back in India, and then Lan Huang, a collaborator at University of California, Irvine. Lan's group is really the world's expert in this technology called cross-linking mass spectrometry. And so what Kamal worked with Lan and Clinton in her lab to work out is a way, can we develop a technology to combine our expertise in cell surface proteomics with cross-linking mass spectrometry, with the ideas that we could use these homobifunctional chemical cross-linkers to ideally lock surface proteins at the cell surface in situ in their native conformation. We can then isolate and enrich for these proteins using the biotinylation approaches I told you about, and then apply this on the mass spectrometer, identify crosslinks, map these back to structures in the protein data bank, and try to find crosslinks in the PDB that we find in our experimental data set that don't match distances in the PDB, suggesting that that resting conformation in the PDB is not what's present on the tumor cells. And so I'm running a little short on time, so I won't go too much into details. I'll just say we found a very promising hit related to another integrin, integrin beta 2, that we hypothesized based on the mass spec data was in an open conformation. We validated this on cell lines. I'm showing you primary patient samples here, showing that most were positive with some degree of heterogeneity for this active conformation of integrin beta 2. We worked in this case with Dev Sidhu's lab at the University of Toronto, who's a world leader in phage display-based approaches. We couldn't get our nanobody platform to work in this case, but very fortunate to collaborate with Dev's lab and Jay Adams in his lab. And so using phage display, developing recombinant antibody fragments against integrin beta 2. Kamal found one that was highly potent in the CAR T cell format that we move forward with development. Again, this is just showing, I think I'll skip this movie actually for sake of time. In terms of thinking about that on-target off-tumor tox, we showed that the antigen wasn't present on normal hematopoietic stem and progenitor cells, so that was very important. We could also demonstrate specificity for activated T cells. Sorry, I'm rushing through the data here. But then what I want to get to is the really important thing in the last minute and a half here, is really mentioning that the really critical test, we found that this active integrin beta 2, we know can be expressed on some hematopoietic cells when they become activated, but most hematopoietic cells should have this in that closed conformation as well. And so what Kamal did was he generated these humanized immune system mice, essentially implanting human CD34 positive hematopoietic stem and progenitor cells into immunocompromised mice. If you wait about 2 to 3 months, they develop a humanized immune system. About 20 to 50% of their circulating immune cells are actually human CD45 positive. And then what Kamal could do is treat these humanized immune system mice with our active integrin beta 2 CAR T, a positive control of a CD33 CAR. This is another leading target in AML that has gone into clinical trials but has known toxicity issues, and then an empty negative control. This is showing you the depletion of human CD45 positive cells a week after giving the CAR. And as expected, the CD33 targeting CAR totally eliminated or largely eliminated those human circulating immune cells, whereas our active integrin beta 2 CAR didn't touch them at all. It looked like the negative control. And then also our antibody is actually cross-reactive to murine cells. We saw from murine complete blood profiling, also no differences after treatment with our CAR T. So it's very promising from a toxicity perspective. We know this antigen is only expressed on hematopoietic cells. So these are the only cells we really have to worry about. And then also, but in terms of efficacy, what was very promising is we could put these into patient-derived xenograft models. Here showing you the Kaplan-Meier curves. Again, comparing the CD33 CAR, which has already gone into clinical trials, we see essentially equivalent efficacy of our active integrin beta 2 CAR in these two different, one less aggressive, one more aggressive model, but essentially equivalent efficacy of our CAR T cells to the positive control of the CD33 CAR. So really quite promising that we may have developed a new antigen for this tumor with great need that has a very strong toxicity versus safety profile. And so it's something that we also want to move forward into clinical translation. So just getting to the final slide here in terms of take-home messages. Really want to illustrate that mass spectrometry-based proteomics, we think, is a powerful tool for immunotherapy target discovery. Identifying these protein-level alterations, particularly in the context of what I illustrated for conformation, but there may be others, may have specific potential for discovery beyond just looking at protein abundance, and particularly beyond transcriptome-only analyses. Again, but even after target discovery, you still have a lot of work to do this on-target validation, preclinical testing before moving to translation. So that's what I want to leave you all with. Just say thanks. I mentioned the folks in my lab who've worked on this along the way. And again, thank our funding sources and collaborators, and happy to take questions during the Q&A. Thank you all.
Thank you very much, Dr. Vita. As a reminder to our audience, you can submit your questions at any point for any of our addressing any of our speakers at any point during the webinar. and we'll try to get to as many as possible during the Q&A session that follows the presentations. Our next presenter is Tulia C. Bruno, who is an assistant professor in the Department of Immunology at the University of Pittsburgh and a faculty member in the Tumor Microenvironment Center and the Cancer Immunology and Immunotherapy program at the UPMC Hillman Cancer Center. She obtained her PhD in immunology from Johns Hopkins in 2010 and completed her postdoctoral fellowship at the University of Colorado in 2015, both with a focus in tumor immunology. Bruno has built her independent research program around understanding intratumoral B-cell and tertiary lymphoid structure function in multiple human cancers. And her overall research objective is to develop a B-cell specific immunotherapy in the next 5 to 10 years. Bruno is actively involved in the UPMC Hillman community and is an advocate for women in science, as evidenced by her current role as chair of the UPMC Hillman Women's Initiatives Task Force and her contributions to the Society of Immunotherapies Women in Immunotherapy group. Everything looks like it's up and running. I can see your slides, and it is in presentation mode. So please, Dr. Bruno, take it away.
Thank you so much. Hopefully you can hear me okay as well. So I just want to say thank you so much for the opportunity to present here today. I'm really honored to be among some really amazing scientists, and actually both speakers set me up nicely, both from the technology standpoint, but also thinking about, adoptive T-cell therapies and CAR Ts and how that fits into what I'm going to speak about today. So I'm going to talk about B cells and tertiary lymphoid structures, and it's probably of no surprise to anyone who knows my research program that that's what I'm going to discuss as a new forefront for cancer immunotherapy and how to harness those. I'll start off by saying that we're going to come back to this idea of an immature, mature TLS throughout the talk. It's not the end-all, be-all. This is a big kind of active area of research, I would say, in the field, thinking about the types of structures that are within the tumor microenvironment and how do we properly harness them. But just to give you a visual of what these look like, these are two snapshots in head and neck cancer, actually, that show both a less well-formed TLS and a very well-formed TLS. And usually the hallmark of that is formation of what looks like a germinal center, which you can see here right up against the tumor. So we're going to talk a little bit about this today. And the other thing I want to mention is that even though I'm going to focus on B cells and TLS in particular, I was a T-cell immunologist and still am a T-cell immunologist, hence my tagline. And so I think that it's really an orchestration of all these cell types that's necessary for new targets and for cancer immunotherapy moving forward. If there's questions we don't get to, I've left my email here for you to see. Okay, let me make sure this goes forward accordingly. Maybe we're stuck. There we go. Okay, so I don't have to introduce this too much, but basically there's a lot of different immune subsets in the tumor microenvironment that we need to think about, and there's actually non-immune subsets in the tumor microenvironment. So I think we need to think about both of them. And historically, we focused on the T cell, and I think cancer immunotherapy is focused on the T cell, and that makes a ton of sense. It's the cell type, particularly CD8 T cells, that goes after and kills tumors. It has the effector mechanism. A lot of the immunotherapies that are have gained a lot of traction, such as CTLA-4 and in particular PD-1 and some others that are coming online like LAG-3 have really set the tone for reinvigorating those T cells. But the reality is that there's a lot of other cell types in the tumor microenvironment, not just immune cells and tumor cells. There's also stromal components that dictate a lot of what's going on. We'll talk about that a bit today. And there's a lot of different immune cell types. So today I'm going to talk about the cell types that are within TLS and how we can harness those. And if I have time, I'll talk a little bit about how that dictates B cell function as well. And so our goal in the lab is to really look at the complete tumor microenvironment, particularly in relation to cellular neighborhoods like tertiary lymphoid structures or TLS, and understand how to make better therapeutics for patients. And really the reason that is because we think about, and to take a quote from my dear friend Jen Wargo, we think about it, cancer, regulation or the immune response against cancer as an orchestra. And so if the T cell is the soloist in the middle that's performing, we would argue that B cells and tertiary lymphoid structures could be that help that really helps the T cell to perform and do well. And so that's the analogy I often use, and Jen has used that before. And so I told her I stole it, and I'm going to continue to use it, but I give her credit for it. So that's fair. The other thing I think that's really grown in the field and given us traction, and this is from Dr. Karen Wooler-Gallo, actually. She presented this at our recent Keystone meeting that we had, showing growth in our field. And I think this really demonstrates why we as a field are getting more and more excited, because there's a core group of individuals who have been working on this for a while, but you can see the paper growth over the years for TLS specifically. And so I think that there's a lot of data out there, and I'll go through some of that today, demonstrating why these are important in the tumor microenvironment and why we should be thinking about them more. Okay, so just some background before I get into some data is what are the hallmarks of a TLS? So for those of you that are not familiar, these are what we use in the lab through a multispectral imaging platform. We use the Koya Motif platform actually to do this, to look at TLS differences in tumors. And so you get zoning, and you can get zoning, not all the time. And so that's something that's a bit different than what we see in secondary lymphoid organs, but you can see zoning and borders for B T cell borders, particularly CD4 T cells. You get high endothelial venules via PNAD. And so that's been a hallmark for a very long time for these ectopic structures. And then we use CD21 to start to mark the presence of a germinal center or a more mature structure. And those are going to mark your follicular dendritic cell networks that you can see there in kind of a cyan color. And we also look at activity. So we look at Ki67 and AID to look at lymphocyte proliferation, but also somatic hypermutation of the B cells. So these are some of the hallmarks that we think about, but I think that we're trying to, as a field, come together and put forward markers that will give a consensus for these structures moving forward. So you'll see images like this throughout the talk. And so one of the issues, though, even if you think about bucketing TLS into immature, mature, there are different types. And I think we're finding this more and more now with spatial transcriptomics, especially, and that's something I'll talk about a bit today, which has really helped us propel this field forward and think about TLS in patient tumors. And so this was from a book chapter that my previous graduate student, Ayana Ruffin, put together, who's now a postdoc in Crystal Palace's lab at Emory. And what we thought about was we have this immature kind of paradigm that we've established, but the reality is there's a spectrum, and there's actually an earlier structure that we call as a field a lymphoid aggregate. And there's a little bit of a debate as to whether or not these are always early structures or structures that are maybe on the end and collapsing. So we're thinking about that more, but for simplicity purposes, we think about early structures as lymphoid aggregates. We think about these in the middle as different types of immature TLS. And then we think of these mature TLS as those that have more of a secondary follicle-like structure that has particularly germinal center B cells and memory B cells and plasma cells associated with it. So these are mature TLS with the germinal center. And really the reason that this composition varies is due to the microenvironment. And we'll talk a bit about that today. And so some take-home messages that we'll go through, I think, throughout the talk are, first, that cellular neighborhoods are helpful. I hope that I can at least convince you of the fact that, for the most part, in solid tumors, having cellular neighborhoods is a helpful thing. Now, as we get more and more granularity on what types of cellular neighborhoods there could be those that are functioning better than others, et cetera, but that's something that I think we're learning as a field. The tissue matters, so where these structures form is important. The microenvironment, not just the tumor microenvironment, but the base tissue microenvironment can play a role. Not all B cells are created equal, and that's because B cells tend to gravitate to TLS if they can, but they can also sit outside TLS. And so if I have some time at the end, I'll show you some data from a paper revision that we have ongoing that kind of demonstrates this a bit in head and neck cancer. And then I think we have to keep in mind that even though we take cues from secondary lymphoid organs and also from other disease sites where we see these ectopic structures like autoimmunity or viral infections, that the tumor is going to dictate some of this, and it's never going to make things that are canonical. And so that's something we always keep in the back of our head. And so some clinical data that has been summarized really nicely in this review in JX Med actually a few years ago now, and probably needs an update, is that there's multiple cancer types where both B cells and TLS have been shown to be prognostic. The larger the dot means the more, how many patients were analyzed, and the darker the green, the better prognosis. And so you can see there's several solid tumors that this has been demonstrated for B cells, but also for TLS. But really what I think got the field, particularly the cancer immunotherapy field, really excited about TLS was a series of papers that came out in Nature in 2020 that showed that in kidney cell melanoma and soft tissue sarcoma, that TLS were prognostic, or not prognostic, prognostic is probably the wrong word. They were associated with patients that did better on immunotherapy. And in fact, the group that worked on the soft tissue sarcoma, so Wolf and Catherine Friedman, they actually then went on to conduct a clinical trial where they bucketed patients just by TLS. So they entered them into the trial by TLS. And this was with Antoine Italiano. They showed that and demonstrated that when they did that, the response rate in those patients was even better than the bucket trial. So I think that there's plenty of clinical evidence that these TLS can help us understand patients that might predict or respond rather to immunotherapy. However, there's still a lot to learn there because there's different ways that TLS can be modulated. And so that's really when we started to think about how TLS play a role in solid tumors and how we can study them better. And more importantly, how do we maximize their function in the microenvironment? And I think the other reason that we're really interested in this is because TLS, going back to that initial picture, you'll notice that CD8 T cells aren't present in this panel. And that's not because they don't become involved within this process. It's because the reality is that they don't often get into structures. Sometimes they can, and we're trying to understand that better. But really, they often flux into the tumor better as a result of having a structure there. So even in some of the mouse models that we're working on, when we see structures, we see better influx of CD8 T cells. But certainly in human studies, both from Jen Wargo's group and others have shown that when you have CD8 T cells flux into the tumor, they also have better activity. And we've actually shown in head and neck cancer that when you have TLS, particularly TLS that are more sophisticated, that the CD8 Treg interactions go down. So I think that lends a lot of kind of evidence to why these structures could be important in orchestrating responses within the microenvironment. The other complication that we have, which I'll touch on some of these today, is that it's not just the cell types that are hallmarks of structures. There's a lot of other pieces involved with the formation of these structures. So they not only need the right cell types, which we've already gone over through this image, but they need initiating factors. And these cytokines and chemokines have been shown pretty richly, both in secondary lymphoid organ literature, but also in tumors themselves. And so I highlight these because these studies have been vetted in cancer. And so these initiating factors are quite important in cancer, although sometimes some of these initiating factors don't always just come from the same place that they do in secondary lymphoid organs. They could come from immune cells as well. So CXCL13 is a good example of that. And you need antigen. So there was a lot of talk about antigen in the last talk. So you need the right antigens. We've shown in head and neck cancer, when there's HPV present, that the virus really dictates the type of B cells that are there, but also the type of structures, having more mature structures when there's HPV present. We've done some studies with Tim Han and Abby Overaker here at Pitt, showing that the microbiota can also regulate TLS formation. And so that's been in a mouse model and now following up in humans. But really, I think immunogenic cell death in general can help get the right type of antigens that allow for these structures to form and to thrive. And then finally, I think the stromal tumor axis is very important. And we as immunologists often ignore that, but it's quite important and we need to think about that. So mesenchymal stem cells, and they can be precursors to fibroblastic reticular cells or even to follicular dendritic cells that we marked here with CD21. And they can regulate what's happening with TLS development because TLS thrive on stroma and they have to have the right stroma in order to thrive and stay. And we know that the tumor can educate the stroma in a negative way. So before I talk about just one vignette from the lab to kind of demonstrate some of this, I wanted to just show you some of the imaging we're doing across tumor sites, just to show you the variability of TLS in different solid tumors. And so we have here a little schematic that we put together showing a lymphoid aggregate somewhere in between an immature TLS, which I explained to you that there's probably several levels of this, and then a mature TLS at the very end that has more of a sophisticated germinal center. And so we use tonsil controls as a positive indicator of what a structure would look like. And we don't really call these TLS, these are secondary lymphoid organs. And you can see really nicely this really nice structure that's forming. You can see zoning, you can see FDC networks, all of that within this tonsil. This is our control. And then I'm going to bring up all the other tumor types that we've started to look at. And I don't have time to talk about everything today, but I just wanted to paint a picture that it's very complex and it's different. And it's not just different across solid tumors, but it can be different within the same solid tumor. So for example, colorectal and head and neck cancer, they often have more mature structures. And in the bottom right corner, this is the number of patients that usually have structures and potential drivers of these differences, some of which I've already alluded to. And non-small cell lung cancer, we've started to think a lot about the primary lung. We have a lot of studies in this in humans and mice, as well as thinking about brain metastases from these patients. And you can see that it's quite different when it moves to the brain, which is probably not surprising. Even in melanoma, we've shown differences based off of site. So the lymph node looks different from the skin, which looks different from brain metastases. And what I'm going to focus on in ovarian cancer today is how site-specific differences might drive TLS, particularly when you compare either the fallopian tube to the ovary or the ovary to the omentum. And so I'll talk about that today in more detail. So this just paints a picture of the different levels of complexity that we see in solid tumors. And that's why we really look across tumor types, not just within one, because we feel there's a lot to learn when we cross compare. Okay, so getting into the ovarian cancer story. So this is captained by a postdoc in the lab, Ian McFawn, and he's done a really nice job using a lot of new spatial imaging and transcriptomics to really understand TLS in ovarian cancer patients. And we got interested in this because a lot of the studies in the ovary are really looking at structures in the ovary or the omentum and not necessarily looking at other sites of disease like the fallopian tube. And so Ian really wanted to understand what TLS formation looked like across. So he used that panel that I showed you earlier, and he created a machine learning algorithm to quantify TLS, which was a big thing for us because we often rely upon pathology to do that, which is still important. And we work very closely with our pathologists. But this allows us to machine learn and count structures in an unbiased way. So here you can see every green island is a TLS, basically. And then we can quantify. And the inset shows a more granular view of those structures. And so you can see even from the inset, and I'm going to show you some data to demonstrate this, that the type or the way the TLS are put together and structured are also different. So not only does the fallopian tube have a lot more TLS than, like, say, the ovary. Here you only have one green island, and there's some patients that their ovaries have nothing. But also the composition as well as the maturation state is going to be very different. And of note, we quantified against benign ovary and fallopian tubes, and those don't have any TLS. So when there's no cancer present, you don't really see a lot of TLS in these tissue sites. So this was the first thing that we did. And then we took that and we could do outputs for composition of the TLS, as well as what type of TLS is there. Is it a lymphoid aggregate, or is it an immature TLS, et cetera? And so to show you some of the composition, we first compared the fallopian tube to the ovary, because those seem to be two large extremes for us. We're still working on the omentum. And we also compared it to stick samples, which are serous tubal intraepithelial carcinoma, which are precursors in the fallopian tube. So depending on your school of thought, you either believe that disease starts in the fallopian tube and then migrates to the ovary and the omentum, or you think that it could start in the ovary and then spread accordingly. It's been shown pretty definitively that, in the fallopian tube, you can go 7 years and it could develop over time, and that starts as a stick and then can develop into a full-blown tumor. So we're still trying to understand this, but we use this as a comparison because this would be an earlier precursor lesion. So these are the data looking at the cellular outputs of common immune cell types that we would think about. And there's a lot of data points here because each of these is a structure. But basically, ovary TLS have decreased B cells, but they also have increased CD8s and Tregs. And that probably makes you think for a second and pause, because I just told you that CD8 T cells don't often get into these structures. But we're seeing differences in CD8 T cells in the ovary versus the fallopian tube. And so one thing that we think could be happening here, and we're following up on, is that CD8 T cells and Tregs actually can make CXCL13 and sometimes lymphotoxin beta, which is different than in normal lymphoid organ structures. And so we think that potentially those cells are there because those ovary TLS, which I'm going to show you, are less sophisticated. So they might be calling for the B cells to get in to form the structures and calling for that help. That's one hypothesis we have there. The other thing we noted, which probably explains the decreased B cells, is that we looked at HEV numbers as well with the panel that we had. And when we looked in the fallopian tube versus the omentum versus the ovary had far fewer HEV than the other two sites. And so when we looked at the nearest neighbor analysis, which I'm not showing you here, the HEV really like to interact mostly with the B cells. Out of all the lymphocytes that are shown here, the B cells are the highest interactors with HEV. So that could be another reason why those structures have less. So there's a few different things with composition that we're seeing. And the reason that this is important is because Ian then went back to the TCGA to say, well, if I look for, in a large data set, if I look at the B cell signature and look at its prognostic value versus if I look at a 12CK signature and its prognostic value, or the combined signatures, how do patients do? And so the 12CK signature is going to be the vetted signature for TLS in the field right now. We're trying to fine tune that as a field now that we do have spatial transcriptomics, but it works well. And you can see that both of those signatures on their own have prognostic benefit for ovarian cancer patients, but it's really when you combine the two that you see a better benefit. And so this goes back to my initial point, was that it's not just one thing. There's multiple things that need to come together for these structures to be present and to work well. Okay, so that's the composition. What about the maturation? So not only can Ian machine learn, and actually the rest of the lab, because everybody uses this algorithm for the TLS, which you can see here, but they can also machine learn for FDC networks forming a germinal center. So then that gives us our criteria here below where we score the structures. So 1 is going to be your lymphoid aggregate, 2 is going to be some sort of an immature TLS, and then 3 is going to be the more mature TLS with the germinal center. So when Ian used this output to look at the fallopian tube versus the ovary versus the omentum, he actually saw that the ovary had more lymphoid aggregates and less structures that had germinal centers compared to the fallopian tube and the omentum. And that's probably not surprising based off of everything that I've already shown you, but we were able to machine learn and quantify that. And not only can he look at that, but he can use the Ki67 and AID for proliferation and affinity maturation of the B cells and demonstrate that within the germinal centers, when they have germinal centers, this is infinitely better than if they don't. Now, there's still a range. And so this is where we start to think about what type of germinal centers are there? Are they early germinal centers? Are they incomplete germinal centers? And that's something we're following up on. But we do know when the FDC networks are present and we see a germinal center, we have better B cell activity, both from proliferation and affinity maturation. So all of these imaging data are great, but what we wanted to do was then weave our imaging data with spatial transcriptomics because as was mentioned in the first talk, it's really revolutionized a lot of what we can do, particularly with FFPE tissue. And we gravitated to the DSP because they were set up to work on FFPE tissue. We also like the DSP a little bit better than the 10X because it allows us to go in and select TLS on a whole tissue slide, which wasn't the case when we first started that. Now 10X is starting to do that for whole tissue slides. And then we can also segment, which is something that was really important to us. So we can look at, within that structure, we can look at B cells separate from other immune cells, separate from tumor, and then we can do negative selection with the segments for stroma. And I'll show a little bit of that later. And so there's a lot of readouts for this platform in case you're not familiar, but most people are. And what I'm going to be showing you today is the 18,000 gene WTA readout. And we have started to do some cosmic work for this. I'll show you a little bit of a teaser data at the end if I have time. Okay, so what we did was we then went back and we looked at the ovary, which is here on the top, and the omentum. And so you can see here even the differences with structures for size, et cetera. And this is showing you the morphological staining that we use and then the DSP segmentation that we use. And it's showing and demonstrating that we can segment on these different populations. We can select the TLS, we can select adjacent to the TLS, et cetera. And what we first wanted to do was ask, what is a TLS DSP signature? Can we derive one? And the reason we wanted to do that is most of these signatures have been either hypothesized from single cell suspensions or hypothesized based off of what we know in secondary lymphoid organs, but they have not been derived directly from TLS in situ. And so I think a lot of people are doing this for multiple different solid tumors, not just us. And so when we did that and we asked that question just about TLS regions versus non-TLS regions, we saw that TLS regions had these genes most predominantly expressed that you can see here in blue or in the TLS regions compared to the non-TLS regions that we selected. And then what Ian did was he went into the TCGA and he vetted the survival benefit of this TLS DSP signature in the TCGA cohort versus the 12CK signature. And he saw that it had prognostic benefit, and it was really very much pronounced in people that were under 55, which is around the time when you go through menopause. So this would be considered earlier stage or earlier patients, etc. So that was also interesting to us, and we've been thinking about that more. But it does vet for survival, which is great. The other thing that we did is we wanted to see if we could recapitulate what I was showing you with the imaging data for TLS trajectory, or basically different types of TLS, but with spatial transcriptomics. And so what you can see here are TLS features: immature, intermediate, or mature, or no TLS. And so what we saw was this trajectory that you can see here, and you can see the colored dots are going to be more no or immature up here, and then it goes toward mature and intermediate. So we found this intermediate TLS signature, which was not present in our imaging data. So this is why the transcriptomics helps. And we also noted that in looking at the ovary versus omental TLS, that it followed suit with what we saw with our imaging data, that the ovary had more immature TLS and the omentum had more mature or intermediate TLS. And so what we see when we look at imaging is then we put in this intermediate TLS, and you can see that it is somewhere in between here and here. And so we're now following up on this trajectory and understanding the genes that govern these different steps. So this is one thing that has allowed us to uncover differences in these trajectories and genes that might govern it. We were also able to look at the CD20 segment, specifically within either a more mature structure or a less mature structure. And so purple is more mature, and you can see that the B cells have different genes that go up. Some that make sense: TNF family receptor proteins, CD79B for B cell signaling, BCL6 as well, which is a hallmark of a germinal center. So those all make sense. But then you start to see things like the CTLA4 CD80 axis that are also upregulated there. And so this could be a regulatory mechanism. It could be that some structures have maturity, but maybe they don't have as much activity. And so this is something that we're thinking about a lot in not just ovary, but also other solid tumors. So what we wanted to do is then understand how the omentum in the ovary with spatial transcriptomics compared to just a lymph node, not a diseased lymph node, a normal lymph node that would have structures naturally. And so it's probably not surprising that the number of genes that are different between mature structures in the lymph node versus the omentum are less than between the lymph node and the ovary, because I've already told you the ovary have structures that are less developed. And so the gene sets are much higher. But what I found interesting was that the top genes, once you reach that maturation status, are very similar in the two sites, and they're associated with actin mobilization and metabolism, not surprisingly. And so what we really wanted to do was dig into this question of immature versus mature. And so what we did was we looked at our imaging data, we characterized immature versus mature, and we went back to our spatial transcriptomics and bucketed those patients. And we saw that mature TLS had a very clear signature. And you can't see all the genes here, but some of the genes that were very interesting to me are here that really focused on focal adhesion, motility, cell matrix adhesion, basically, genes that would suggest that you're interacting with some stromal compartments, or you need to interact with some stromal compartments. And I mentioned to you that the stroma is quite important. So we went down the angle of thinking about the stroma. And so we did this in partnership with Lon Kaufman. She's a very talented physician scientist here at Pitt. And we started our labs around the same time and naturally gravitated toward each other because I was working on the immune system and she was working on mesenchymal stem cells, which can very much regulate the immune system in tumors. She mostly works on cancer-educated mesenchymal stem cells, which express WT1. They've been shown to be very immunosuppressive and promote tumor initiation. They correlate with worse prognosis compared to bone marrow derived or normal mesenchymal stem cells, which also migrate to inflamed areas, particularly the bone marrow. These can be tissue resident, the normal ones, and they can be precursors of things like follicular dendritic cells, which are important for TLS formation, as I've already shown you. And when we looked at her TCGA, she had done some bulk sequencing on these different these different mesenchymal stem cells. And we saw certain factors, such as this platelet-derived factor that is really important for FDC differentiation, was much higher in MSCs versus cancer-educated MSCs. And then we also vetted, and she's shown this pretty extensively, that WT1 is very much upregulated on these cancer-educated MSCs. So that's what we use to mark those structures. So what we wanted to do is take a step back and look at the stroma in a very unbiased way. And so Ian used his spatial transcriptomic data, and he used his stromal segment for TLS adjacent stroma versus TLS distal stroma. And then we looked to make sure that our segmentation worked. And this is hard to see, but what we looked at was stromal pathways versus immune pathways. And there is a little bit of immune contamination because it's not 100% perfect, but I would say over 85% of that stromal population, and they're the top pathways that we see, with mesenchymal stem cells in that list as well. So we were on the right track. So then Ian went and he just asked a simple question, was if we looked at the TLS adjacent stroma and we looked at the TLS distal stroma, what does the chemokine expression look like? Because that's one of the places where a lot of key chemokines come from. And you can see here that the adjacent stroma expresses a lot more of the chemokine and chemokine receptors that are necessary for TLS development compared to those that are distal. We also looked at WT1, as I already mentioned, that's a pretty good hallmark in the stroma for cancer-educated mesenchymal stem cells. And we saw here that it was increased in the distal stroma compared to the adjacent, but I think even more convincingly, it was one of the top pathways that came up in the distal stroma compared to the adjacent, particularly data sets that are WT1 targets. But to really hammer this home, we used Lon's CAMSC signature, which is a 13 gene signature that she has published, showing that in the adjacent stroma, there's a fewer signature for CAMSCs compared to the intermediate versus the distal stroma. And we vetted this a bit with spatial imaging. We now have a combined vector panel or motif panel where we can look at the MSC types in relationship to the TLS. So we're working on that now. But just by doing simple confocal with an adjacent section, you can see here's the TLS, and you can see all here in yellow, those are bone marrow derived MSCs that are very much they're much closer to the TLS compared to the purple, which are the cancer-educated MSCs, which you really don't even see there. So we're vetting this further. And we've rolled this with some in vitro data, looking both at FDC differentiation, but how these different mesenchymal stem cells influence B cells. So this is just one readout that we've done. We've done others, but just to give you an example, we've developed these in vitro assays because Lon can isolate the MSCs from primary patient samples, and then we can ask how well do B cells adhere to those, et cetera. And so you can see that the normal MSCs hold on to the B cells much better than the CA-MSCs. And that's not surprising given some of what I've showed you, but we've been able to vet some of the biology that we've seen spatially in vitro in a dish. And then finally, to end this part, and then I'll wrap up and just say a few more things about this topic, is that the stroma was also the stromal signature that I mentioned earlier, the 13 gene signature for CAMSCs, we wanted to pair it with our TLS DSP signature. So we went back to the TCGA and we showed that if you have high TLS signature and a low CAMSC signature, that's when you do the best. And if you have a high CAMSC signature, it almost negates completely the effect of the TLS DSP signature. So indicating that this also affects survival as well. So now what we're doing is we're taking our spatial transcriptomic data sets, not just in ovary, but we have a whole data set in lung, and we have head and neck, colorectal, and melanoma that are being basically worked on now, all for TLS and cross-comparing to try to unify TLS signatures or see if they're different. And also think about pre-detection and pre-malignancy. I mentioned the stick samples in ovary, but we're thinking about a lot of other pre-malignancy settings as well. And we also have an overt effort with mouse models that we're thinking about in our lab, but also in concert with a team grant that I have for the Mark Foundation with these investigators listed here. We call ourselves Team TLS, where we're looking at multiple different tumor types and also mouse models to understand TLS. So to wrap up, what I would leave you with is that these very complex immunological hubs are going to be important, I think, for IO, but there's still a lot to learn, and we need to understand in different tumor microenvironments. And we can use spatial transcriptomics and imaging to get at that. That's really revolutionized what we're doing. But we also have to think about crosstalking human data and murine models. We are also thinking a lot about how this B-cell T-cell crosstalk might be dictated by TLS formation, and also the function of B cells might be dictated by TLS formation. And the last thing is that even though these have been correlated with better response to immunotherapy, particularly CTLA-4 and PD-1 or that combo, we have to look at how certain therapeutics modulate B cells and TLS, because I think that will help us understand. And so if getting a TLS makes better CD8 influx, that could make better sensitivity to PD-1 blockade. And there's been some papers in the last year that has actually indicated just this in some mouse models. We are thinking more on the upfront end, how the tumor-stroma-immune and even the microbiota interface is regulating these structures. And I would say that we often thought about immature versus mature TLS, thinking that mature was always better. And there has been studies showing both in our lab and others that having more mature TLS is better, but it could just be that these have differential function. And so while we think about the type of TLS, we also should think about the activity of TLS. So I'm going to just take 2 more minutes, because I think I have the time, to talk a bit about cellular neighborhoods and what they mean and how they could dictate B cell function, and how we're trying to understand this both in humans and mice. So just to give you a little teaser, this is a busy slide, but in lung cancer, we have both human data sets, but we also have mouse models that form TLS naturally over time, both in a carcinogen-induced model, but as well as in that orthotopic version of that model. And we're now trying to modulate TLS via an oncolytic virus and delivering certain factors that we're finding necessary from our lung cancer data sets in humans to modulate TLS. So this is one way that we're going to get at how these cellular neighborhoods are modulated and the mechanism. The other way we're doing it is looking at spatially what's happening with the TLS. And this is in head and neck cancer, and work that was captained by my previous graduate student, Ayana, that I already mentioned, and taken over by our new graduate student, Allie Casey. And what we've demonstrated in head and neck is that based off of where the B cells are, they can go through different pathways, and they can actually also have differential function. And so this is showing where the B cells are related to a germinal center or to an extrafollicular differentiation. So extrafollicular differentiation requires TLR ligands and cytokines and different cues. And what we saw was that B cells that were outside of TLS or within TLS without a germinal center really participate in this pathway versus a germinal center derived pathway. And the reason that's important is that germinal center derived B cells, particularly switch memory B cells, they have better functionality than extrafollicular derived B cells, which are these double negative populations that I don't have time to go into, but they just don't work as well. So this is another angle that we can think about, maybe modulating the B cell function, right, in relation to the TLS, or modulating the TLS in relation to the B cell, and trying to understand that. And so the cellular neighborhoods will dictate this. And so one small look into the cosmics that is going to help us with that is that you can see these different niches that come out of the cosmics data, because now this is truly at a single cell level, where the other spatial transcriptomics are not. And you can see that the B cell signals are high in these different niches. And we would probably categorize these 3 niches as more immature, earlier TLS. But what you're seeing, if you look at these broken down, that some of these have more fibroblasts and macrophages compared to others. Or if you go on the other extreme, structures that look more mature, you can see cell types pop out that we might not have thought about, like plasma cytoid dendritic cells. So I think pairing the single cell dynamics with what we've already done with spatial transcriptomics is going to be really helpful for TLS and really understanding the different interactions and the different types of neighborhoods that are present. So with that, I'll stop and leave you with this cartoon that we put together a while ago, thinking about how B cells and TLS could be modulated. There's a lot of different ways we can think about it, a lot of different places in the cancer immunotherapy cycle that you could think about where these could be harnessed. And I think that the field is really thinking about current therapeutics while we try to discover new therapeutics that might be synergistic, particularly, like I said, maybe targeting the stroma is the key to help out with some of the therapies that we need. So with that, I'll stop and thank my lab. I have a really amazing team. I turned 40 in July. They threw me a big birthday party. You can see us on the right-hand side there. I want to thank all of my collaborators. I mentioned some of them by name, but I didn't get to talk about everything today. I'd like to thank my funding, as well as all the patients and their families, because we really take a back translation approach in the lab. We think about what's happening in people, and then we develop the right mouse models to really study this. And now we're going back and forth between the two. And so thank you for your attention. And if you're interested in what you saw, we have a consortium that meets monthly to talk about B cells and TLS. I'm happy to sign you up if you email me. It's virtual and it's free. Thank you so much.
Thank you very much for that presentation, Dr. Bruno. Our final speaker today is Ed Roberts. Ed Roberts completed his PhD on cancer cachexia at Cambridge University with Doug Fearon, and then studied tumor dendritic cells during a postdoctoral fellowship at the University of California, San Francisco with Max Krummel. Roberts is currently a junior group leader at the Cancer Research UK Scotland Institute, where his lab investigates how the tumor influences T-cell priming in the tumor-draining lymph node. Let's just make sure everything's up and running from a technical perspective.
Looks good. If I do that.
Yes, looks great. Yep.
Okay,
Perfect.
cool. If you hear a thud, I've fallen asleep. I've been up for quite a long time now, but I'll try and stay awake. So thanks to everyone who's gone before me. I think they've given a really good grounding, which is going to make some of my introduction incredibly redundant, but we'll speed through it and get to the data. But I guess what I really wanted to talk to you about today was about what our lab's really thinking about, which is thinking about the tumor microenvironment and how really we believe it extends out to the tumor draining lymph node, and how that's then influencing the research that we're doing, looking at the interactions going on there and how they influence response to immunotherapy. So I'm going to start with the most overused slide ever, but just sort of point out that why are we talking about this? Well, and obviously from Jim Allison's Nobel lectures, we have the amazing responses we get with sort of checkpoint blockade and some immunotherapies where we can get essentially functional cures in a subset of patients. But the aim is to work out how can we expand this to allow that benefit to a broader array of patients. And one thing that's been really fruitful, I guess, in approaching this has really been thinking about how T cells develop. And one of the things that's really sort of been obvious is that as T cells develop, we sort of move from naive T cells through sort of initial priming in the lymph node. They move into these niches where you have these T progenitors, precursors of exhausted cells, which can then, surrounded by CDCs, they then become exhausted T cells. But as T cells become activated, they upregulate these checkpoint molecules, for example, PD-1, TIM-3 later. And so as a result, a lot of focus has really been about how to block these checkpoints. And as a result, a lot of the research is focused on what goes on in the tumor microenvironment. And this has been very successful, and it isn't, without reason. And apologies for the pixelation. But for example, there's many studies, but I just thought I'd highlight this one from Siddiqui et al., which came out in 2019. And it's a bit of a complicated diagram, so just focus with me on in panel F, you've sort of got this solid black line. I don't know if you can see my cursor, probably not. But the solid black line is how the tumor would grow normally. And then you look at the green line and you've got checkpoint blockade. And the key thing about this is that this is occurring with the addition of FTY720. And now, just for people who may not know about this, FTY720 is a drug that blocks lymph node egress from the lymph node to the tumor microenvironment. So essentially what this shows is that in the absence of cells moving from the lymph nodes, you can get reactivation of T cells in the tumor microenvironment, and this is sufficient to control the tumor. And then that's just sort of repeated in G, where you can see tumor weight. And now the red line just shows that the cell that seems to be important are those precursors of exhaustion, precursors of exhausted T cells. And this sort of led to that paradigm where, checkpoint blockade can really unleash these precursors of exhaustion. They can differentiate and attack the tumor, and that's great. And this seems to all make sense of what's going on in the tumor microenvironment. The complication of this is that there's also a series of of other papers that have shown in different mouse models that there can, in some of these models, be a requirement for the tumor draining lymph node and what might be going on there. So I just thought I'd highlight this one from Franson et al. And in this, they sort of, again, we're looking at PD-1 blockade, but what you can see is TDLN resection. So the tumors were put in, the tumor draining lymph node was resected, and then checkpoint blockade carried out. And what you can see is that in the absence of a tumor draining lymph node, the tumor was not controlled by PD-1 anymore. And this is shown in A by looking at tumor size, in B looking at survival, and C in a different model. So, in C, the model was whether you resected before or after, and essentially, if you resected after treatment, you still had a poor response. So what this suggests is that in some models, at least, you needed an intact lymph node in order to have a response to checkpoint blockade. And then this was really shown in a really beautiful bit of work from Joachim Eert's lab in the Netherlands. And the first author, Floris, did a really beautiful set of experiments where they actually they had sort of a lung tumor model, and then they delivered the checkpoint blockade intrapleurally. So rather than going for systemic checkpoint blockade, they delivered very small amounts of the checkpoint inhibitor into the pleural cavity, which then drained to the lymph node. And there's obviously a lot of control showing that this is where it went, but what they saw when they did this is that intrapleural injection, which is the sort of the blue bars at the bottom, led to reduced tumor weight, increased CD4s and CD8s, and that this was obvious, and this was blocked if they added that FTY720 again. So showing that they could get really solid responses by just delivering that drug to the lymph node. And then even more excitingly, they had access to a lot of melanoma and matched lymph nodes, and were really looking to see whether this could be relevant in patients. And so they developed an assay where you could see you could see when PD-1, PD-L1 interactions were occurring. So wherever there was a co-occurrence of those two molecules, you got staining. And they then looked at the number of these contacts, and then split patients, so recurrent-free survival based on sort of less than 48 months or more than 96. And essentially, those who recurred faster had more of these PD-1, PD-L1 interactions within their lymph nodes. So while there was sort of some conflicting data about the importance of the lymph node in sort of mouse models, this really seemed to highlight that potentially there was a role for the lymph node in sort of the prognostic outcome for patients. This was also then sort of highlighted by some work that suggested that, while we've been looking in a lot of these mouse models, which are obviously very rapidly growing, and the length of time that mice are treated is relatively short. In patients, this can be a much longer period of time. And what seems to happen is, if you look at in the sort of panel on the left, what this was looking at was TCR alpha and beta motifs in the tumor and in the tumor draining lymph node at 8 weeks and 17 weeks. This is in a longer mouse model. And what you could see is that the motifs stayed shared between these two sites, suggesting that T cells were continually being repopulating the tumor from the lymph node. And then when you look on the right, this was then looking at clones in patients after pre and post treatment. And what was found is that there was a huge expansion of many novel TCR clones. So what this sort of potentially suggests is that while there is this continual repopulation from the lymph node, which now has also been shown in other ways, including using KD mice, where you could label cells and look at that migration back and forth, that seems to be important potentially in patients where over a longer period of time, those precursors of exhaustion are unlikely to provide sufficient long-term cells, and the lymph node seems to be coming into play. So now I'm going to take a step back, now that hopefully I've got you on board with thinking about lymph nodes a bit, and sort of point out that one of the really big problems that our lab was interested in is, well, how do you know the kind of response that you should drive in the lymph node, and how does this go wrong in cancer? And one of the big problems we were trying to formulate, and we ended up falling down on the sort of the area of information theory, which is that the challenge the immune system has is you have some immune challenge, be it a virus, a tumor, whatever, even homeostasis and the requirement to maintain tolerance. And this all can occur in a tissue, which is spatially distinct, is spatially separated from that lymph node. And the lymph node is also draining a field, so it's getting a whole bunch of signals coming in from all over. Now, somehow, a source message, which is that specific challenge, like a viral challenge, needs to somehow be transmitted to the lymph node in a way that can then lead to effective and appropriate T-cell responses. And so in this model, in the traditional information theory, you need the source message which needs to be encoded, transmitted, received, and decoded. And what we're thinking is the specific challenge somehow has to be encoded. And this usually gets done by dendritic cells. And the sort of obvious thing is that we've got dendritic cells that sit in the tissue. They sample whatever the challenge is, whatever's going on. And based on the types of signals they receive, that dictates how they process the antigens, the surface markers they put up, including CD80, 86, and other co-stimulatory markers, and the cytokines they produce that will direct T-cell response. They then can migrate directly to the lymph node, and there they can interact with T-cells, which can receive those signals that were encoded earlier. And this then drives T-cell differentiation, which is essentially them decoding that original source message. Now, this all seems really, really tidy, especially when we consider these migratory dendritic cells. But the really big problem for us was that those migratory dendritic cells aren't the only cells that are carrying out T-cell priming. So within the lymph nodes, you have other dendritic cells, which are lymph node resident. So these have never been in that peripheral tissue. They've never seen that challenge, and yet somehow they must respond appropriately. And again, the real problem being that multiple different areas of tissue are going to be draining to that same lymph node. Some are going to require one response, and other areas will require a different. And somehow those resident DCs have to be able to pass that information. And from what we know is they can't receive the signals in the same way that the T cells do. So we somehow need we needed to square that circle. Now, what we do know is that migratory dendritic cells can transfer antigen to resident dendritic cells. And this was shown by a bunch of really talented people from Frank Carboni's lab in Australia. And then we later, during my postdoc in Max Crumwell's lab, showed that they could sort of synaptically pass antigen between them. But we don't really know how that's being regulated. But then we know that those resident cells help direct T-cell fate, but we just don't know how they would know how to do that. So to get at that, what our lab's been using is an approach where we use a very stable, bright fluorescent protein called ZS green, and we express it under different conditions. And then what that allows us to do is look at that antigen source. We can then look at any sort of immune cells that ingest some of that green fluorescent protein, or the ZS green, because they become very bright green fluorescent. We can then look at how they've encoded that by looking at their cell surface and the phenotype and what they produce. And then we can track it to the lymph node and look at the T cells interacting with them and start asking, well, what does that mean for the T cell phenotype? So first of all, we've done this before in tumors. I haven't presented it here, but we published it from Max's lab a few years back now. I I think 2010 was the second one. But once I started my lab, I knew that we wanted to compare this to a different setting, so that we would actually be able to look at how that encoding differs based on the type of challenge. And so if you want to see more details of it, we actually published it slightly earlier this year in Science Immunology in the references down there. But I just thought I'd go through it a little bit now, which was that we started by taking influenza A virus and cloned downstream of the non-structural protein 1, a self-cleaving 2A peptide and ZS green. Now in B, you can see that this didn't actually stop the virus from growing in vitro, which was pretty fortunate for us because ZS green is quite a large protein and flu does not like proteins being added into it. But what this allowed us to do was then to image where that infection went as well. So we infected mice, and at day 3 or day 6 post-infection, we inflated the lungs with agarose and imaged them by light sheet microscopy after clearing them. And what you can see is at day 3, you have these very bright fluorescent airways where you've got the infection localized there. And by day 6, it sort of starts to spread out into the parenchyma. So having modified the flu, we haven't really messed with how it's spreading. It looks like a normal infection. And when we imaged thick lung slices, again, that's on the right, we could find these areas of green, and we could find immune cells there, and we could even see them picking up that material. So here we've just looked at neutrophils. So we'd sort of established a model to look at now a viral infection with this, where we could track its antigen, and we could compare it to our previously published tumor model, which in this case we used a B16 melanoma that expressed ZS green that we could inject IV and then look at how it colonized the lungs. So again, this allowed us to look at 2 challenges, both draining to the mediastinal lymph node. And then just to cut a very long story short, if we did this experiment, if we looked in the lung and looked at sort of CDC1s in the top and CDC2s at the bottom, again, for people who aren't familiar, it's really not that important for today, but CDC1s tend to be better at cross-presenting. And we tend to think of them as being more involved in CD8 priming than CDC2s, but that's a very loose definition. But in the lung, what we saw is PR8, which is our flu. When we looked at the antigen-bearing DCs, so these are the ones on the green line, they massively upregulated CD80 and 86, both the CDC1s and 2s. Whereas in the tumor setting, they barely upregulate CD80 and wasn't even statistically significant, and they do upregulate CD80, 86, but much less. So then when we go to the lymph node and we look at the resident dendritic cells, although the activation is much less, we actually see this pattern repeating. So for the flu, we see upregulation of CD80 and 86, and for the tumor, we only see upregulation of 86. So somehow, the antigen is being brought from the periphery, carried to the lymph node, and once it's transferred to these resident cells, they're somehow knowing the appropriate way to become activated based on the peripheral challenge. And so we wanted to understand what this could be. And again, long story short, there's a few things being proposed. For example, T-cell help, sort of local secretion of cytokines. None of these made much sense to us because, for example, T-cell help requires the dendritic cell to present antigens agnostically until another activated T-cell activates them further. And that sort of loses some of that exquisite sensitivity that dendritic cells have of choosing how to process antigen based on its origin. And sort of secretion of cytokine didn't allow this tight activation of only the antigen bearing. So we ended up looking for other possibilities. And what we came to was thinking about, could you co-transfer material? And obviously, we know that things like toll-like receptor toll-like receptors can sit inside phagocytes, and these are important for activating dendritic cells. So when we looked in this flu setting, we could find the ZS green inside phagocytes, but we could also find double-stranded RNA and obviously a TLR ligand. But when we got to the resident DCs, we saw that those were being co-transferred. And we did some more stuff in cancer, but again, you can read it. But then the big final experiment was we made mixed bone marrow chimera mice, where we had a small proportion of the bone marrow derived cells, so about 15 to 20%, were unable to respond to, were MyD88 knockout, i.e. they couldn't signal through some of their TLRs. Again, broad brush. And when we did this, we could look in the lung and we saw that normally you get a certain amount of activation in the MyD88 sufficient DCs, but this was blunted in the MyD88 deficient ones. Now, crucially, 90% or 80 to 90% of the migratory dendritic cells that pass the antigen are going to be MyD88 sufficient. So if it was that they're secreting other signals, they'll still do that. But when we went to the lymph node, we found that actually the resident dendritic cells needed MyD88 themselves in order to become activated upon receipt of the flu material. And so what this sort of led us to was this, sorry, I'm distracted by little emojis floating up the screen. What this led us to was a model where we sort of came to the conclusion that the periphery and the lymph node was highly integrated, and that you get co-transfer, at least in flu, of TLR ligands, and that this was important for the full resident dendritic cell activation, and that contextual clue allows a lossless information transfer system and maximizes signal to noise. Now, why does this matter for cancer? Because I know that's what you've all tuned in for, and I have been wittering on about flu for a while. But what we could also see is that, for example, multiple groups have shown that the dendritic cells that differentiate in the tumor microenvironment acquire a regulatory phenotype, and we could see this being transferred to resident dendritic cells in the lymph node, where they didn't upregulate some of these co-stimulatory markers, but they did upregulate things like PDL1, which nicely relates back to the paper from Joachim Ertz's lab, where they showed that most of those PD1, PDL1 interactions were between DCs and T cells. So we believe that this tight transfer is leading to that sort of regulatory or immune suppressed phenotype to move from the tumor to the lymph node, but in an antigen specific way, which then can explain some of the observations about how a tumor draining lymph node can still support an immune response while still having poor response to that tumor. But this obviously raised the big question for us of what does this sort of partial activation of lymph node resident CDCs actually mean for anti-tumor T-cell immunity? And the reason this question is so pertinent is that while we know that resident DCs have a role, it's been very difficult to unpick the role of the different DC subsets in the lymph node because of they're not very genetically tractable, especially to separate the migratory from the residents. And we just haven't really had brilliant models available. And so what we were interested in is what's going on here. And this was really pushed a little bit more by a recent paper by Prognevska et al., which was this sort of beautiful paper where they compared LCMV to two different tumor models and looked at how T cells differentiate in that lymph node. And so for those of you who aren't familiar with these, cell trace violet obviously dilutes from right to left as T cells divide. And then they were looking at sort of PD1 and other markers of this. And what you can see is that when you have an acute viral infection, you rapidly upregulate PD1 and downregulate CD62 ligand. You downregulate TCF1 and you upregulate TIM3. But when you look at the priming going on in these two different tumor models, while you do still upregulate PD1 to an extent, you don't downregulate CD62 ligand, and you don't upregulate TIM3 or downregulate TCF1. So there's obviously something going wrong with the priming. And we wanted to understand if this could tie to those different DC functions. So to get at this, people before have obviously introduced a lot of these ideas, but we sort of looked at high dimensional immune fluorescence. So we basically ended up building, based off of Gary Nolan's paper, the Codex paper, kind of built one out of Lego and attached it to one of our confocals. and have been sort of building up a panel to look at all of these different subsets that we're interested in within the lymph node. Just because of time, I'm not really going to go through it, but we've been exporting these into Fiji, building a sort of an open source pipeline where we go through CellPose to do our segmentation, we go into QPath to export the polygons, and then we can take those polygons into sort of FlowJo to do normal kind of flow cytometric assignment of cell identities, which we then use in RStudio to sort of perform a linear discriminant analysis. Again, don't need to go into that for now, but it's given us a really nice way to identify all of our different cells and then to look at them as polygons so we can start looking at cells that are interacting. And what this has really allowed us to do is infect our mice with flu to start with to understand, well, what happens during a good response. And what we see is, I'm only going to show data from day 6 actually here, but we took flu, we infected, and then when we've done these images, we can identify our different CDC subsets. What we can then do is look at the CD8 T cells that are contacting those different T cells and ask about how they're activated. And one of the markers that we've been looking at is ICOS. And the reason for this is that when we separate our granzyme B positive and negative T cells, ICOS is really the marker that we could find that was a cell surface marker that was upregulated as you acquire actual effector function. So when we looked at the T cells that were in contact with these different DC subsets, the main thing to see is that the migratory CDC1s and resident CDC1s both have upregulation of CD69. And CD69 is an early marker of T cell activation. There are other things that might cause it, including cytokine, but this was kind of what we were seeing, is that those T cells that were in contact with these CDC1s seem to be being activated. And this is reassuring because those are the ones that have been suggested to be important for T cell, CD8 T cell activation. But when we looked at ICOS, apart from in one of our lymph nodes, we've generally found that this was activated specifically in the CD8 T cells around the resident DC1s. So this suggested to us a model whereby while migratory DCs can activate T cells, you need these resident dendritic cells to give that full commitment, full activation. To test that, we tried to take the reductionist system where we took our ZS green expressing flu, but this time we're expressed synfecal, which is recognized by OT1 T cells. We sorted out the ZS green positive DCs and mixed them with OT1s and left them 3 days to look at what happened. And as you would predict based on that previous slide, both the CDC1s from the migration and resident could drive T cell proliferation. So we get this expansion with both the green and the blue bar, but we only see those T cells gaining full effector function with the resident dendritic cells. Now, again, I promise you there is a reason why we look at flu, but so what it looked to us is that we need those interactions with resident dendritic cells to make the full effector function, and this happens during a flu infection. So what happens during cancer? So we looked again in these lymph nodes, and here you can see those 4 different subsets of DCs and look at their location. So in pale blue is your B cell zone of the lymph node. And when we look at where the DCs are, they're all sort of huddled into that T cell zone in flu. Whereas when we look at the tumor draining node, we do around the periphery of the T cell zone, we still find migratory CDC1s, but the resident CDC1s are fully excluded. And if you then look at the bottom, this is a very busy graph, but essentially we're looking at the interacting partners of the migratory and the resident CDC1s. And the main thing to take away is that pink is a CD8 T cell. And so you can see even at rest, there's quite a large proportion of migratory DCs that are touching a relatively large proportion of their interacting partners are CD8 T cells. And this doesn't change a huge amount with flu. It does increase a bit, but not a huge amount. And with the tumor, again, we still have those interactions. Whereas the resident DCs are largely excluded from the T cell zone in homeostasis. They obviously relocate during flu, but they fail to relocate during tumor. So what we're failing to get is those clusters which can give full T-cell effector function in the tumor lymph node, which we normally would see during a robust T-cell response. So we wanted to really understand why that might be, and we went back to that sort of our model of this partial T-cell activation, and we know that one of the important signals to move into that T-cell zone is CCL21 that's received by CCR7. Now, migratory DCs sort of constitutively express this. So if you look at the MDC ones, you see that they're basically all expressing CCR7. But if we then look at the antigen bearing versus the antigen non-bearing, we find that in the resident DC ones, upon receiving the antigen, these start to upregulate CCR7. And that fails to happen in the tumor. So part of that poor activation of the resident DCs is this failure to upregulate CCR7 and to relocalize into the T-cell zone. And indeed, this is just quantified on the right. So we then sort of showed that yes, during flu, we get flu and tumor, we can get perfectly good proliferation, but we get less good expression of granzyme B and of ICOS, especially. And so we gave something that we knew would lead to upregulation of CCR7 and relocalization, which is just giving them poly-IC. And when we put this subcutaneously, what we could see is that the OT1s that we transferred that would recognize the tumor suddenly did start expressing this ICOS in the tumor draining lymph node. So this is sort of work that we're just sort of building up now. So any comments very welcome. But it leads us to this sort of model that we have, which is that normally our migratory cells come into the lymph node and they can start driving T-cell proliferation, but they also pass antigen to these resident dendritic cells, which upon receiving it move to the T-cell zone because they upregulate CCR7, and there they can allow full T-cell activation. But when we come to the tumor setting, although we get the antigen to the lymph node and the migratory DCs can cause that T-cell proliferation, they pass antigen to resident DCs. Those resident DCs fail to upregulate CCR7 and so don't relocalize. And as a result, we end up with these T-cells being produced, which don't actually gain full effector function within the lymph node. And so even before those T-cells hit the tumor, they've failed to become the most effective tumor killers that they could be. And so I guess just going back to that, the sort of graph that we had earlier, the diagram we had earlier, sorry. So we have that naive T cells turn into these sort of stem-like T cells. And this is sort of from that Prognostica paper and from our data, showing that these T cells, even though they start to differentiate, they upregulate PD-1, they don't downregulate TCF7, they don't upregulate interferon gamma, they don't turn on granzyme B, and they move to the tumor in this sort of partially activated state. There they can interact with tumor CDCs and become these precursors of exhausted cells, and there they gain effector function. And then they can go off and become these exhausted T cells. There's an added layer of complexity because these precursor exhausted cells can actually move back to the lymph node. But the main thing to really focus on, at least for us, is that while there's been a huge amount of focus on what can go on in the tumor microenvironment, one area that's been sort of neglected to an extent is really thinking about what could we strategically do to improve priming in the lymph node. And obviously this is now coming to the fore with sort of a lot of vaccination approaches and seeing how those can synergize with some of the responses. But what we believe is that seeing as we've already got quite a lot of this antigen in the lymph node, if we can really understand what is going on with this programming and why we're not getting fully effective T-cell differentiation in the node, rather than needing to sequence each person's patient and develop these personalized tumor vaccines, we might be able to just sort of reprogram the node And ultimately, the lymph node is the best GLP cell producer that we know. So it would be great if we could get that to do some of the work for us. And with that, I just want to thank sort of members of the lab. I've put up a few of our undergrad students and master's students who, for some reason, dragged us to do the flying trapeze for Christmas. But also, I just want to thank our collaborators in the School of Immunity and Immunology and Infection, so Georgia Perona Wright especially, and then our collaborators over in the Center for Virology Research, because without them, we really wouldn't be able to modify all of these sort of viral challenges in the ways that we need to look at them, or at least we wouldn't be able to do it without accidentally releasing it or doing something awful. And that is that for me. Thank you.
Thank you very much, Dr. Roberts. And I'd like to extend a thank you to our whole panel today for sharing your work with us and allowing us to, I think, learn new things and gain a better perspective of what immunotherapy looks like now and more importantly, can potentially look like in the future. At this point, before we jump into our Q&A session, I'd like to take an opportunity to once again thank our symposium sponsors, ATCC, Biorad, Canopy Biosciences, Sinobiological, and BPS Bioscience. Dr. Lunenberg is unfortunately not able to be present with us today or at this point because he had to attend to some other business, but we do have our other three presenters on. Before we jump into our Q&A session, a final reminder to our audience to feel free to submit your questions for the panel at any time using the Q&A tab on the right side of your screen. And I think before we talk about anything maybe too specific, I noticed a trend across the presentations that studying cancer is now involves more data volume, more data points, multiple layers, multiple ohms than ever before. So I wanted to ask the panel their own kind of thoughts and thresholds on sampling practices, how to judge whether data is representative or not, and whether that changes depending on the technology used, the situation, the modality, whether it's multi-omic or single-omic, so on and so forth. To get our discussion started, I'd like to ask Dr. Vita to respond first.
Yeah, sure. I think from our perspective, in terms of target discovery, thinking about kind of a multi-omics approach, really, again, we're focusing on using proteomics as an initial target discovery. Many of those times they're on cancer models as opposed to directly on patient samples for the discovery-based approaches. And so that is something that for us, it's a really big step in terms of if we find something that we think looks promising from a proteomic perspective, we compare it to publicly available transcriptional data sets to say, hey, we think this has a good profile in terms of on-target off-tumor tox. The next step is really kind of that one-by-one validation on patient samples. And so really that involves collaborating with our other oncology colleagues here in pediatric and adult oncology and having access to banked patient specimens, and then flow cytometry really one by one comes down to the key steps that we need to go from kind of that omic-based discovery to actually homing in on a specific target. So I say for our lab, that's really the common progression.
Thank you very much. Dr. Roberts?
I think similarly, we use a lot of, we're very fortunate, and well, most of our work is in mouse at the moment, at least. So we've actually been fairly fortunate with some of our imaging approaches to be able to sort of use things like tissue clearing and to actually just image entire lymph nodes, for example. So we haven't really come up against the problem of having to worry about our sampling being representative, where we've been able to look at whole tissues. I think sort of going forward, where we are gaining access to more patient samples, I think really the only thing we can really do is just make sure that we sample enough and keep examining the results of that sampling to see how, are we starting to see sort of subsets within there, or are we sort of coming to a more stable view as we add more things? But to be honest, it hasn't been something that we've had to worry about too much, because we've had the luxury of whole lymph nodes and whole lungs, for example.
Thank you very much. And Dr. Bruno?
Yes, so I think that, I just to complement, I think what the other two speakers had said, we have access to a lot of patient material here. And I think that at UPMC and Pitt, and I think that what's really kind of helped with really understanding what's going on, particularly with TLS with large data sets, are the technologies that I showed today. So basically, having a really good spatial imaging, having really great spatial transcriptomic readouts, and now moving on to the single cell version of that, I think is going to be very powerful. So then in that way, we can request retrospective patient samples via FFPE blocks to really do our discovery-based approach that then allows us to solidify the targets of interest related to the TLS, and then vet that both in mouse models, but also then go back and do validation in independent patient cohorts. And sometimes that requires doing flow cytometry and fresh specimens. So we also have banks of samples for that for certain tumor types. So that's helpful. But some of that's also prospective collection, or like, for example, combining with cellular biology or cellular immunology, showing you some of what we do with the cancer-educated mesenchymal stem cells. Those come directly from the patients. So I think it's this multifactorial approach that allows us to do discovery and then vet the biology. And then I think what's going to be really important in what we're starting to work on now, which I didn't get a chance to talk about today because it's still something that we're curating, is really looking at well-annotated samples for patients that have been treated to really understand how treatments modulate the tumor microenvironment. But specifically for us, we think about B cells and TLS. And so we've been fortunate to design very smart and well-controlled clinical trials with our clinician partners here, not just in ovary, but in other tumor indices that I didn't get to talk about, to really collect the samples, to do that vetting and really look at pre-post changes, which I think is important. So those would be the things that I would add to what's already been said.
Thank you very much. Our next question is for Dr. Bruno. Are there any solid tumors that are not associated with TLS, whether immature or mature?
So I am yet to see that there are solid tumors that don't thrive as a result of having structures. I think that the overall message from particularly that review that captured a lot of this nicely was that having structures, regardless of the state, is better than not having them. There are a few tumor types, and these are not data that have been published, that there might be trends sometimes in the other direction, but it's not common knowledge that that's happening. And those could be very specific tumor types that have very unique microenvironments, even more unique than what I showed today. But like even immunosuppressive environments, very immunosuppressive environments, like pancreatic cancer, for example, there's been a lot of studies showing that if you have TLS, you do better regardless of the state. So I haven't seen anything necessarily in the opposite direction, but that doesn't mean that it's not going to come out, especially with all the different data sets that we can acquire and all of the different technologies that we can utilize to really look at this in a granular way. I think as we dig into this more, we might see some different correlations down the road. But as of now, to my knowledge, most are prognostic in a favorable way.
Thank you very much. Dr. Vita, you talked about... Sorry, let me start. So surface receptor expression is very dynamic, and it's something that you alluded to in your presentation as well. How do you navigate that factor of trying to make sure that you're not capturing a snapshot, but also at the same time to try and understand what causes a receptor to be present or not present. And I think the question can also be extended to conformational shifts as well.
Yeah, I think just great question. And overall, one thing we can lean on right now, at least, is we do have data from clinical trials and FDA-approved products for CD19 targeting CAR Ts, BCMA targeting CAR Ts, and many other investigational. And really, what ends up happening is there are mechanisms of so-called antigen escape or antigen negative relapse, where some tumors, whether it's selection of antigen low tumors, whether it's genomic deletion of the locus encoding the antigen, whether it's mutations at the specific epitope that the antibody and the therapeutic binds to, these mechanisms have been described. They typically seem not very common. It's usually a minority of patients exhibit these type of antigen negative or antigen escape relapses. So there can be some dynamics there, but it appears to not be the major mechanism of relapse. Is much more thought to involve kind of the, at least in the CAR T field, phenotypes of the T cells themselves becoming exhausted, otherwise non-functional over time, is probably the major mechanism. But we can't rule it out. It could change for different targets. Different targets have different biology in terms of how they distribute to the surface, be recycled. And it does vary from product to product, even against the same target. You can see different mechanisms being more common in terms of antigen negative relapse. So that's clearly something that it's hard to assess even in the models that we have available. The vast majority of modeling, as we did, is whether patient-derived or cell line-derived models in immunocompromised mice. It really doesn't predict the resistance mechanisms that you're going to see in a human for the most part. So it's difficult to know a priori before you start the clinical trial. For the conformation selective antigen, we do think this is definitely more of a concern, is can the tumor essentially go back to that closed conformation and avoid the therapy? At least in our patient-derived xenograft models so far, again, we haven't seen that as a resistance mechanism in the murine models, but it could happen in vivo. I think for that context, ideally, one thought is you find, if you find a conformation or antigen in general that the tumor is biologically dependent on for proliferation and survival, maybe it's less likely to be able to lose the antigen and still proliferate within the body. So that's one thought, is there some selection disadvantage if they lose the antigen? But again, it's something I think we'll have to see when we get to the point of trials. Essentially, is the tumor able to change this conformation of integrin beta 2? It may be receiving some important survival signaling through that pathway.
As a follow-up to that, is that a possible avenue for relapse as well?
Yes, absolutely. It could be. Exactly. So I think that's one of the, I think that's certainly something that we will look out for and to stay tuned and talk about, but we are figuring out how can we make other multi-targeting CAR T something that's other folks in the field have done. But we think this target in particular, I kind of only showed it very briefly, but we do know there is some heterogeneity in patient expression already, where some patient tumors may not express a lot of the antigen. So can we combine this with other targets that have shown a similar phenotype where they probably are safe but have heterogeneity in the tumor? Can you make a multi-targeting CAR T that still will be safe, but can eliminate tumors that express one or the other? And that might be the best therapeutic. So that's actually the avenue we're going toward for clinical translation.
Thank you very much. Next question for Dr. Roberts. Does the impact or the influence of the lymph node, the dendritic cells in the lymph node, does that differ across different types of cancer, across solid versus blood cancers, for example? Or is it fairly uniform?
I think it's a really good question. I think that there'll be, caveat this is going to be mainly speculation because I'm not sure that it's really been looked at. But for example, at baseline, the types of dendritic cells that you have in, say, the gut differ from the types that you get in other tissues, as does the sort of the organization of the lymph node and some of the things going on there. So I would be very surprised were there not to be some cancer type specificity. When you start getting into leukemia and lymphoma, lymphoma, you have the added complexity of the sort of the leukemic cells interacting with the stroma of the lymph node and starting to alter the lymph node function as a whole. So I think that there's a huge amount of nuance that is there to be discovered, but really our understanding of what's going on in the tumor draining lymph node is really not well developed compared to some of our, I mean, admittedly, there's obviously a lot more to learn about the tumor microenvironment and structures like tertiary lymphoid structures, but our understanding of just what's going on in these different secondary lymphoid organs is fairly poor at the moment.
Thank you very much. As a follow-up to that, what is the role of the lymph node for exogenous T cells?
So it's an interesting question. So there was some work done by Miranda Brose back in, I think, 2014 maybe, where transferring in sort of adoptive cell transfer of pre-activated tumor-specific T cells in a mouse model didn't require anything from the lymph node. So adding in FTY720, you got as good of a response. But again, this has been done in mouse models based on our understanding of sort of the precursor of exhausted cells and the lymph node being able to maintain them. It's hard to say, but I imagine that were you able to bring the T cell in the lymph node, into that to continue expanding to maintain a sort of stem-like or a memory-like population, you would imagine that you would get longer engraftment of these cells. But again, that would rely on us understanding more about the fundamentals of T cell priming and how to do that exogenous priming in such a way that they're able to interact with the lymph node. And again, I think at the moment, how we stimulate these TILs exogenously is usually fairly blunt, and it is improving, but how we're going to get that to work in collaboration with the lymph node is hard to know at the moment.
Thank you very much. Dr. Bruno, I think your presentation talked a lot about TLSs in different tissue types and different cancer types. Is there heterogeneity in TLSs within a singular location? Are there certain environments within that singular location that TLSs form or don't form preferentially, or is it rather more of a uniform distribution?
So I think it's a great question. So I think the comment I will make about this is first that I think we're still trying to understand this better. There's definitely differences across tumor types, which is what I kind of showed with that comprehensive slide. But then if you look in certain tumor types, depending on site, which is really the ovarian story that I told today, it will basically, it can change based off of where the tumor forms. Now, what you're getting at is kind of location and probably proximity to the actual tumor itself. And so I didn't talk about this today, but the field as a whole, and we in the lab, we always look at where the TLS are in relation to the tumor. And they tend to sit on the peritumoral border, but there are occasions when they get into the tumor bed. And when they get into the tumor bed, though, they often gravitate toward the stromal pockets in the tumor bed. So locationally, I would say in general, they always try to associate themselves with stroma, whether they're inside the tumor or on the peritumoral border. And then we're doing a broader look at where they are in relation to other types of structures that could be present. So for example, in the lung, a good example is that you can have airway associated structures called IVALT, for example. And so that complicates things, but that's closer to the airways, right? And so that allows us to look at that in comparison to what's happening in the tumor. And what I will say in the mouse modeling we're doing, where we can look at TLS formation naturally, is that we see that the structures will move and gravitate closer to the tumor as the tumor forms. So I didn't mention this, but we feel like the tumor is dictating a lot of these differences, both in composition state of the TLS activity. And so that's kind of the next wave, I think, is thinking about how the tumor influences that structure formation. But in general, that's a pretty good rule of thumb as to where they're going to locate and be both in human and now what we're seeing in mice.
Thank you very much. As a follow-up to that, does proximity to a TLS affect the tumor cells in any way?
Yeah, there's definitely been studies shown, and the way people have been doing this, I think, by imaging is looking at coding of tumor cells by IgGs or IgAs, and there's definitely evidence that that occurs. And we have shown that the plasma cell readout can be different depending on the type of TLS is there, and that can also be related to the tumor, but those data are not quite curated enough yet. But I would say that the actual tumor itself, I think the field is still trying to understand exactly the mechanism, right, or the function. And I kind of alluded this at the end. The hypothesis that we currently have in the lab is that these higher level structures that have like more of these germinal centers, that they are going to allow for antibodies that could be IgG1 that could help engage NK cells and participate in ADCC or potentially even macrophages for ADCP. But those are more the minority. The more majority are these immature structures that I mentioned. And we think that those are really important for B-cell, T-cell crosstalk that then would allow for a presentation of antigens, particularly antigens that are necessary to educate the CD4 T cells and then offer help to the CD8 T cells. So I think what we're still trying to understand is that direct link potentially between either the B cell and the CD8 and the TLS and the CD8 and how it might be affecting the tumor in patients. But people have certainly started to look at either how the tumor regresses or pathological response if it's a clinical trial to understand how the structures relate to the tumor. But I think that there's still more to do there.
Thank you very much. Dr. Vita, when it comes to selecting a target, whether it's a receptor or a conformation, what is more important, abundance or ubiquity? Is it better to have a little receptor on all of the tumor cells or a lot of receptor, but maybe on 50, 60% of the tumor cells?
Yeah, it's a really interesting question. I mean, I think overall, ideally, you would have that ubiquity in terms of at least you have the chance of eliminating all tumor cells depending on the potency of your targets. I think that is, but then the drawback is oftentimes, at least for the targets that we're aware of now, if you get to that point where all the tumor cells express it, but there's a wide variation in the target density, there's more chance some essential normal tissues will also overlap at the low end of that range, for example. So that's really where the challenges come in. And I kind of alluded to it in the last question you asked, but that is why some of these approaches, whether it's kind of being called in the field logic gating approaches, where essentially you can have so-called OR-gated cell therapies, as I refer to, where you could have two separate antigens that might have good off-tumor expression profiles, but there's heterogeneity in the antigen expression on the tumor. Can you combine the power of those two and still have something safe and effective? There are other ways around this too. If you get to that point of you have an antigen that is uniformly expressed on tumors, but at varying density, and it is at low levels on normal cells, can you come up with so-called OR-gated strategies, which will only enact the killing function, and gated strategies where you only enact the killing function when both those antigens are present and you don't have normal tissues that have both antigens at enough abundance to activate, so you can get the selectivity in that manner. Others have tried to develop not gating strategies where maybe you need to identify one or two antigens on the tumor cell, but then there's another antigen that we know is on an essential normal tissue. And if that one is recognized also by the cell, then you can essentially turn off the killing activity. Those have proven more powerful to engineer, but there have been recent advances in those spaces. So there are ways that essentially trying to turn these engineered T cells into small computers to enact a cytotoxicity program. So there are potential ways to address these issues of heterogeneity, again, either by combining across multiple antigens or if it's ubiquitous, but also in normal tissues, trying to turn off the T cell at the appropriate times. But it is challenging, and especially for these logic gating approaches, really haven't had much yet in terms of clinical data to see how well they really work, but there are trials either in the planning phases or have started dosing patients but haven't released any data yet. So I think we'll get some more information in the future about how those strategies are panning out.
Thank you very much. I think we've got time for one more question, and we started our Q&A session with a bit of an open-ended question. I think we'll close with the same kind of open-ended question. The other big theme I've noticed across all the presentations today is kind of a rise in machine or computational-based either learning or analysis, ranging from software analysis of software image analysis to development of proprietary algorithms, and how that's not whether I can say supplanting is a bit controversial, but definitely moving into a space that was once occupied by traditional pathology analysis. So I just wanted to know, kind of what your comments on that, the advantages of software-based data analysis, data acquisition, and any caveats that are associated with the rise of automation in this space. Dr. Roberts, please, if you could begin.
Well, so it's interesting. I think that obviously the opportunities there are huge in terms of just the quantity of data that you can analyze, and you can analyze in sort of reproducible ways. I think recently, for example, I saw a really interesting presentation where someone had managed to essentially convert H&E images into sort of pseudo IHC to improve diagnostics in areas where people don't have access to that IHC material, and also just sort of, I guess, so there's things where you can do stuff like that, but then there's also the fact that some of our image analysis just isn't feasible to do by hand in an unbiased way. So it's been really exciting to develop some of these models. I guess in terms of the caveats, you can have some really beautiful code, but if you don't sense check and really go back and then use that expertise in sort of a curating way, you can really get some garbage coming back out of your data analysis pipeline. So I think it has really great opportunities, but you just have to have really, at times, creative ways to ensure that you've got ways to sense check and ensure that you haven't got something unusual coming back out.
Thank you. Dr. Bruno?
So I would say that this is already kind of underway for TLS. There's actually a paper I think that has recently come out from the Friedmans. I don't know if it's fully published or on bioRxiv, but they've basically been showing that machine learning with H&E is not enough, that you have to also pair that at least with a single plex for CD20. And so one thing I didn't get a chance to mention, because I want to make sure we got through some data, not just a primer on TLS, was that CD20 is a pretty accepted surrogate for TLS. And so we often use that as our triage or our single plex in the lab, and I think others do. And so we're finding that at least at minimum, pairing it with the machine learning is necessary so that you don't get, as was mentioned already, anything artifactual. From the spatial transcriptomics standpoint, I think there's a few things that are important there to kind of just hit home to make sure that the machine learning you're doing makes sense and it gives high quality data. The first is the design of that experiment's very, very important. So, our core runs a lot of spatial transcriptomic projects, and I'm the director of that core currently. And we make sure we meet with people to make sure we understand their questions so that we look at their slides. We do workup on their slides to understand how to, form the best scientific question and get the best, outputs from those data. As far as TLS go, we have a series of checks that we do on our data before we do any discovery-based work to make sure that the signatures that are supposed to be there are there at minimum. And then we start to look at the discovery. And the same with the segmentation. I mentioned that we vet our segmentation tool and make sure that the populations we are getting make sense and that it has good clear results. And that's really a partnership with our bioinformatics team. Sunny Bao and her team here, they really do a lot of that for us. And we work with them to make sure that we're getting the signals that make sense. And then we go into the discovery phase. So I think there's still more to do, and there's a lot more machine learning and things that you can do. But I think we're getting there, at least in the field for TLS.
Thank you. And to close, Dr. Vita.
Okay, and I'll make it fast since I know we're over time now. But just to say that I think in the target discovery space, the main place we've used it is essentially trying to develop new computational strategies to integrate the proteomic data we're gathering with existing transcriptional databases of expression across other tissue types, predictions of whether certain cell surface markers are actually at the cell, prediction of whether specific genes are actually located at the cell surface in terms of their protein product. I think the main challenge there is that we do have to rely so much on transcriptional data sets. And again, this uncertainty is just because a gene is expressed, just because it's predicted to be the cell surface, is it really there? We can't tell localization, for example, in epithelial cells, whether something's basal or luminal, that could have impacts of potential toxicity or non-toxicity. So I think there's still just limitations on the input data that we can use to draw conclusions from the computational analysis. But it is still an important step. And to say too, there are many different ways to try to find optimal targets. There are a lot of different computational approaches to use it. There's no one gold standard yet. So maybe one of those will be developed at some point, but not there yet.
Thank you very much. Thank you, everyone, for your wonderful presentations and for that discussion. If anyone has any further questions, please consider reaching out to the speakers directly. And as a reminder, the symposium will be archived on the scientist's website, and you'll receive an email notifying you when the on-demand version becomes available for viewing. Please make sure to follow the scientist on social media for updates on future events. In closing, I'd like to thank everyone who took the time to join us today, particularly those of you who shared your questions and comments. On behalf of the scientist, I'd like to extend a heartfelt thanks to our panel of speakers, Joachim Lumber, Arun Vita, Tulia Bruno, and Ed Roberts, as well as our sponsors, ATCC, Biorad, Canopy Biosciences, Sinobiological, and BPS Bioscience. Thank you, everyone, and have a pleasant day.